didn't record. Sorry about the no recording thing, but all we did was we summarized that the endocrine system is about communication. Communication, communication, communication. Keyword. What's going on in the endocrine system is that there's a certain number of endocrine glands. There are major endocrine glands and there are accessory, others. What the difference is between the left side and the right side is that a major or primary endocrine gland is one that only secretes hormones. That's its only job in life. It doesn't do anything else except for secretion of hormones. And that list is pretty short. We're going to be going through this list for lab, so you don't need to write all of these down now. Please, please don't, okay? Pituitary gland, pineal gland, both of those are found in your brain. Thyroid gland, you can palpate that right around your larynx, right around your voice box, right? And then down here, the adrenal glands. There's both an adrenal medulla meaning core, which is such a weird thing, right? And then adrenal cortex, which does not mean core, it means the outer rim. So these are going to be the primary endocrine glands you will focus on for this unit. Yes, these ones are the primary ones you'll be focusing on for this unit, except for this guy over here, the pancreas. We will also talk quite a bit about the pancreas through probably end of this week, beginning of next week. These accessory guys, their primary function is not to secrete hormones. Their primary function is something else. But they have glandular tissue that can secrete a hormone. So check it out, heart. How bizarre is that? The heart secretes a hormone. But what's the primary function of the heart? Go ahead, throw that out in chat. What's the primary function of the heart? Boom. Yeah, it's a pumper, right? It's just pumping blood. Oh, and feelings. Yes, Tariq, you got it. Right, it pumps blood. It's like my favorite organ, but it's actually pretty boring in terms of what its function is. It's a pump. It's a really important pump, but it's a pump. But let's think about, since its function is to pump blood, doesn't it stand to reason that that is a really good organ to help monitor something about the blood? Yes, no? And let me ask you this, and we'll come back to homeostasis and all this stuff in it from AMP1 that I hope you were able to retain. But if too much blood gets back into the heart, what do you think your body's going to do? Is it going to love that, or is it going to hate it? L or H? And this is a really dumb question, but I'll be asking stuff like this through the semester to get you guys all on the same page. Yeah, I mean, the body's all about regulating a normal homeostatic set point, right? It wants what it wants. Your body is Goldilocks. End of story. Everything's got to be just right. So too much blood starts getting back into the heart. The heart needs a way to be able to say, nope, not today. And it secretes a hormone in order to try to fix that problem. It's going to try to get the blood volume down using a hormone for that function. It's really cool. Don't write any of that down. It's just an example. Does that make sense in terms of these accessory organs? Their job is going to relate to their function. They are great monitors for things. So for instance, the gonads, their primary function is to produce gametes, right? Sex cells, that's their primary function. But they also are going to be heavily related with hormones to help with that function. Here's the deal. For this unit, as I said, we'll focus primarily on the left and a few on the right, like the hypothalamus communicating with the pituitary gland and the pancreas. But stomach, heart, small intestines. I wish I got to talk more about adipose tissue, but I don't. Kidney and gonads. That sounds like the rest of the semester to me, doesn't it? Cardiovascular function. digestive system, digestive system, digestive system, renal system, reproductive system. So we'll be covering those in more detail later. Should you still know that there are endocrine glands now? Yes. Do you need to know everything about them now? No. Is that clear? Awesome. Thanks, guys. What all endocrine glands have in common is they all secrete hormones. What does the endo mean again? Crin means to secrete. What does endo tell us? Inside what, though? Let's be more specific now. Within, inside what? Not the body. Not the body. We tried to fix that yesterday. Not soma. The answer is the bloodstream. Does that make sense to everybody? Endocrine tells us specifically blood. Yes, no, maybe so? Cool. And let me actually get you guys to a quick review. Hopefully, well, if it shows my face, I'm super scrubby, so we shall see. I haven't opened this yet lately. Come on, open Skeskami. Nope, it showed me. Hi, I'm a scrub. Let's try to do this again. All right, it's probably going to show my face again. So this is my overhead cam, and it can have issues from time to time. 1 second. This is going to show me again for sure. Hi. All right. Maybe now. Aha. Hey, have you tried turning it off and back on again? That's what I continuously have to do to make this thing work. Super weird technology. Ah, come back. And then they did an update that changed the function of my overhead cam, which really irks my cookies. Okay. So here's how we're going to be doing a lot of our lectures this semester, though. I love Chalk Talk. It's how I learned physiology. It's how I studied all through college. So I actually bought a whiteboard, a huge whiteboard, and I did all of my studying off of that whiteboard. And I highly recommend that if you guys have had difficulty trying to learn how to study, how to sit down and put it all into one place so you can actually learn it, get yourself a whiteboard. They are super duper duper helpful. So with our studies, we'll be doing a lot of what's called Chalk Talk. Okay. My professor was literally just chalkboard. There was not a single PowerPoint that he put up all year. It was colored chalk. All right, so let's just remind ourselves of a quick picture, just so we can understand endocrine a little bit better. We got ourselves a cell. It's just a basic cell. It could be any shape, but we always draw them round. What's the name of the fluid inside of the cell? From A&P1, who remembers? What's the name of the fluid inside of the cell? Fluid. You're not wrong that the cytoplasm is the general fluid name. Cytoplasm includes quite a bit. But in A&P1, you guys were learning about the different environments of the body. And there's a particular fluid inside of that cell, which is ICF. Do you guys remember this now? I mean, again, if you didn't remember, who cares? This is part of the review. ICF, which stands for intracellular fluid. Intracellular fluid. Yes, no, maybe so? And just trying to ring bells for everybody. I'll be doing this through the semester. You're going to hear me say, does that ring a bell? I'm going to be asking you that a lot because human brains, mine, I can't hold everything. And I don't expect you guys to know everything that I ask. Don't think that I do. I just hope that we can ring bells because later in life, when you're trying to remember something to help a patient, ring a couple bells and you know where to look up the answer. Does that make sense? Real life is not school. I love school, but I'm quite aware that real life is not school. Now, Out here somewhere, there's bloodstream. What's the name of the fluid inside of your bloodstream? Specifically, I said bloodstream. Yeah, good. This is plasma. Plasma is going to contain some other things besides fluid, but it's like 90+% water. So that's where most of that fluid's at. Well, ICF's not just pure water either. So now in between the plasma and the ICF, there is a space here. Okay, it's fluid-filled compartment. What's the name of that fluid-filled compartment? And somebody did write that up above. It is not ECF. It is not ECF. Come up with another word for it, pretty please. It is part of the ECF, so you're not completely wrong, but we want to get specific right here. Yeah, exactly, Beck. Is it Becca or Rebecca? Which one do you prefer? I'm an abbreviator like crazy. Becky. I will try my best to remember that. That's awesome. Sometimes I might just say Beck. I apologize. You can just slap me through your screen. My kid's name is Beckett. So a lot of times, I just abbreviate the whole Beck. All right, so this is IF, or interstitial fluid. Now, there's the cool thing. Again, we're just reviewing, but it is an important review for the endocrine system. I'll tie it all together for you in just a minute. Pieces of the puzzle, then we put the puzzle together. These two things combined are ECF. Aha, that's your ECF. Does that make sense, y'all? Everything outside of the cell is ECF, not just this spot in between. Everything. You get some ECF, and you get some ECF. Why do we care about this basic structure here? This is all internal environment. This is all internal environment. This is the inside of your body, and this summarizes the entirety of the inside of your body. There are billions of cells, trillions. Every one of them has a space, a fluid-filled space between the cell, and somewhere out there is blood. And how is a cell going to respond to the rest of the body? It's either going to have a direct innervation by the nervous system, or the other way it communicates is via the bloodstream. Does that make sense? Because if I'm going to take signal from this cell and get it to a cell down here, well, man, for most of the body, the only way to do that is to send a signal into the blood, and then it gets transported down to this point. So this, in essence, is how the body communicates. Now, we're going to come back to the chemical signals in just a minute, but we got to try to remember one more thing on our review is, what the heck is this gray circle? What is this outline indicating? What is this anatomical structure right here, the gray part? Again, basic, because it's the beginning of the semester. What is this? Don't overthink this. Yeah, this is all the cell wall. Cell wall is technically in a plant. Cell membrane is in an animal. Cool, no worries. Yeah, that was going to be my next question. What is the plasma membrane made out of? And Sabrina and Melissa, you guys jumped on this, right? This is made out of a phospholipid bilayer. Phospholipid bilayer. Yes, no, maybe so? Everybody remember this? You never know what y'all's background is. So I'm trying to make sure everybody gets on the same page. And I hope that it feels easy to you. But if not, you just know you need to do just a little bit more work to get caught up. So this is a plasma membrane. And the plasma membrane is a peanut butter sandwich. What? Dr. H, Craig, you crazy. Plasma membrane is a peanut butter sandwich. Think of a peanut butter sandwich. You got bread. Can water soak into bread? Yes or no? Hell yeah, it can. Yeah, can water soak into bread, Brianna? Like, just take bread and throw it in water. Water is just going to inundate that entire bread. Yes, no? You're all right, Brianna, no worries. But I want to make sure everybody's on the same page. Now you got peanut butter. You got peanut butter. You got fats. Can water make it through those fats? Yes or no? Can water get through your oil? No, right? So right here, we have what's called hydrophilic. This is water loving. Water. And down here, of course, is hydrophobic, right? So when we go back to our chemistry and we want to review our chemistry, there were multiple types of chemical bonds. There were ionic bonds, hydrogen bonds, and polar covalent and nonpolar covalent. And it's these covalent bonds that make these either water loving or water hating. Just bear with me. For those that hated chemistry, I'm going to hopefully make it a little bit easier. Again, I started with that with peanut butter sandwich. Is that a nice analogy for some of you that might have had a hard time with this? It's really helpful. Hydrophobic means water cannot get through there. You're not going to get water to soak through a peanut butter sandwich. It ain't going to happen. Now, it's a bilayer. And let's think about why it's a bilayer real quick. This was one of my favorite intros to my graduate program ever. All day long. Yep, phobic like phobia, exactly. Phobia is a fear, right? Like arachnophobia, fear of spiders. Acrophobia, fear of heights. So phobia is Latin for scaredy cat. Hydrophobic, scared of water, which is great because, again, one of my favorite intros to graduate school was this dude was studying phospholipid membranes and their formation, and I was able to look at how a single phospholipid acts in water. And it's really cool, and it really helps you understand how a plasma membrane is created. So if I put a single plasma membrane into water, this side here loves the water, this side here hates the water. This side here moves towards the water, this side here moves away from the water. I should actually say it's getting pushed by the water. I hope that makes sense. So what's going to be happening to this single phospholipid if I were to place a single phospholipid into water? If this side here is moving towards the water, but this side here is getting pushed away from the water, what's going to happen to it? Imagine the physics of it. Does that make sense? This side here is going, yay, water. This side here is going, hell nah. And it's running this way. It's going to end up going into a spin. Yes, no, maybe so? Which is awesome because we don't have just a single phospholipid. We have billions. And your cells are constantly making new phospholipids all the time too, by the way. So what happens if we take one phospholipid and we throw it in with another phospholipid is this side says, hey, you're my buddy, you like water too. And this guy down here, even more importantly, is like, I need to protect myself from the water, so you line up next to me. These have no chemical bonds between them. They're just lining up. And when you do that, there's a water exclusion zone right there in the middle. Does that make sense? And now, of course, we stack them all together. Water exclusion, water exclusion, water exclusion, water exclusion. You guys get the point. And then we still have an entire side of our phospholipid here that's being exposed to water. This cell membrane goes, oh no, my phospholipids are still being exposed to water. So this whole bridge would be flipping. So how does it protect itself? It lines itself up with another layer. And now we have water exclusion here in the middle and here all around. Does this make sense? It's a really important functionality of the phospholipid membrane. It is not bound together by chemical bonds. It's just chilling next to each other like the surface of the ocean. And it just rolls in this mosaic wave that's constantly changing and constantly evolving. And you can push things through this barrier if the barrier allows it. If the barrier allows it. But what do we know about peanut butter is it's pretty selfish. It only likes other peanut butter. So the hydrophobic center will only allow other fats through it. Does that make sense? Because it's non-polar. This is non-polar. Back and review too, because it will be an important part of this semester. Whereas the hydrophilic side is polar. So this center portion is non-polar. So water, for instance, is very, very polar, okay? It can go into the hydrophilic cell membrane. So it's going to go in here, but then it's going to be rejected and it gets bounced back. You actually have to have pores, but if water is big enough, or if there's enough of an osmotic potential, then water can make it through, but that's stuff we don't want, right? That's tonicity from AMP1. That's going to cause the cell to enlarge or shrinkadink, right? Lysis versus, if you guys remember those words, tonicity, you know, the thing that you guys probably hated, one of the things you hated the most, one of the simplest things actually when taught, right? But students tend to get a real aversion to it. All right, but if I'm a fat, ooh, I love this part of the review. Simple, that you're going to be using through the entire semester. Everything in the universe is trying to go from an area of high to low, low to high. H to L or L to H. Everything in the universe is trying to go. Most everybody, spot on, great, great. Let's just think about it real quick. Let's just think about it real quick. I have a hill. This brings everybody together. That's the only reason why we're doing this. Let me get rid of this. Come on, meow. All right. I have a hill. What's going to be easier? Pushing a ball up that hill or rolling the ball down that hill? P or R? Yeah, down, right? That's easier. And that is what the universe is all about, is saving energy, expending as little energy as possible. Everything's trying to go from an area of high to low. This is called passive diffusion. Yes, no? Or just passive. Passive transport. Everything's trying to go from high to low is going to be passive transport. Diffusion is just an example of this passive how do you spell transport? Transport. You know, it's the easy things I forget. And so if we have a cholesterol or another fat in the system, if I have this, it's going to try to go from an area to high to low. And it can push its way through the hydrophilic ends because it has something that it's really attracted to, which is the core of this. Does that make sense? So it's literally going to push its way past the hydrophilic edges. Yes, no, maybe so? Remember, these are not chemically bound to one another. So this is something that students often this is where they have trouble with. They're like, how can it make it through a hydrophilic barrier? Because it's going from high to low. Then it's going to shove its way past those polar ends. And it's going to party here in the middle. And then there's going to be attractants inside of the cell that cause it to go the rest of the way from an area of high to low. So this is passive diffusion. And a lipid slash fat slash cholesterol, all of these are what's called membrane permeable. They can all just pass right through the cell membrane because they are non-polar. Does that make sense? The peanut butter can match with the peanut butter. These are all peanut butters in our analogy. This definitely plays a huge role in the endocrine function, and I hope that it will help you with the rest of the semester as well. Listen, I do like to throw a couple of definitions down. Polar. This is a word students often have a lot of trouble with, is this whole idea of polarity. Polar because you guys see it with bonds, but polar also because of the nervous. You guys remember those words? Polarization, depolarization, hyperpolarization, repolarization. Polar polarization. No, that's not one. And the way to remember this is a polarizing conversation. Is this bringing people together? Do polarizing conversations bring people together? Not normally, right? They tend to be very abrasive. And that's where the word polarizing conversation came about. Polar means to separate. And that's literally how we can understand polarity. Polarity means to separate. That's the simplest way to understand polarity. In the body, especially when dealing with chemical bonds, what we're talking about is we're separating charges. Now, there are other ways that we can polarize something, for sure. But the primary way we think about polar is to separate charges in the human body. And so a polar molecule, if you guys see this, polar covalent, what do you already know about them? Do they have separate charges or do they have the same charge? Separate or same? SEP or SAM? Or just SE versus SA? And you can get this just from its definition. So when you learn the definition, it really helps you understand a polar molecule better. Bear with me, everybody. I'm only saying what, 1, 2, 3, 4, 5, 6. 6 people just answered that, and there's 22 online. Let's go. Is it same or is it separate? Based on its definition alone. If I see a polar covalent bond, what do you know about the charges? The charges are separated. Good. And this all had to do with that electronegativity, all that crazy stuff that you guys learned about in chemistry. I'm not going to go back through electronegativity. If you want to go through it with me outside of class, just let me know. It's really easy to teach you it, but I don't have time in today's class to go through it. One of the best examples of a polar covalent molecule is water, H2O. And we end up writing this out with a hydrogen that has a bond to a, sorry, I didn't say hydrogen, an oxygen. I did say hydrogen, but you know what I meant. I meant what you knew. An oxygen, hydrogen bound to a hydrogen. 2 hydrogens and an oxygen, H2O. Yes, no, maybe so? Now think of oxygen as something like the sun, where things want to gravitate towards the sun. The sun has so much mass that it's drawing things towards it. Well, oxygen is powerful, super powerful for electrons. And electrons want to spend their time near this oxygen. What charge is an electron? Negative, positive, or neutral? Good, it's negative. And if electrons want to spend their time near this oxygen, what charge is the oxygen going to be relative to the hydrogens? If the electrons want to hang out over here, they're actually going to hang out over here. What charge will the oxygen be relative to the hydrogens? Hold on. What charge did you guys just say electrons were? The electrons are hanging out over here. So what charge will the oxygen be if electrons are hanging out near it? Do you see the difference in what you guys were thinking there? Maybe I said it wrong, I'm not sure. So this is called a dipole, where the electrons hang out near the oxygen, therefore the oxygen tends to be more negative. The electrons are not near the hydrogens. They've been drawn away from the hydrogens. So we're losing the party of the electron around the hydrogen. So the hydrogens tend to be more positive. We separated charges due to the electronegativity of that oxygen. Do you see how this would be polar with a separation of charge? Yes? Again, I can teach you this more out of class if you had trouble with this right now. I went pretty fast. But regardless, can you see that there are charges? Charge, charge, charge. There are positive charges and a negative charges, or a negative charge. So we have separated charges. Yes, no, maybe so? So is this polar or non-polar? Is this P or N? Awesome. And polar molecules are like magnets. Okay, you can think of them akin to magnets. Magnets. Because what's going to be attracted to the negative oxygen is a positive from somewhere else. Does that make sense? Well, what better example of that than another water molecule? And now this hydrogen is going to be attracted towards this oxygen side, and boom, we got ourselves a hydrogen bond, and it's what makes water water. One of the major properties of water. It's polar. Lots of other polar molecules out there, like proteins, for instance. So we'll come back to that. But polar loves polar because all polar molecules are charged. All polar molecules can interact because all polar molecules are charged. All polar molecules can interact. But they hate things that don't have charges. So polar molecules cannot interact with nonpolar molecules. Nonpolar tells us there's no charge. One of the best examples for that is oxygen, because it's just an oxygen that's bound to an oxygen. I should change my color so you guys don't get confused. I'm not talking about polar right now. I'm talking about nonpolar. An oxygen has a double bond with an oxygen. And if I have two of the exact same molecules, two of the exact same elements, which one of those can win in a tug of war for electrons? If they're exactly the same, if I have two sumo wrestlers that are exactly the same, which one wins? They both win or they both lose. Either way, are they going to be pulling electrons more towards one side than the other? Yes or no? And therefore, there's no charges. I mean, that's the power of the nonpolar molecule, is nonpolar molecules are not charged. So when we go back to our cell membrane, the middle is not charged, sides are charged. And that's why water can interact with the inside. Water can interact with the outside of the cell membrane. Water just cannot interact with the phospholipid, the lipid portion, the hydrophobic core. And that's why we have an ICF. That's why we have an ECF. But then there's no water in the middle of those cell membranes. Is everybody on board with that basic idea? Was that a helpful review? I hope it was. You will use this material through the entire semester. So do your best, hold on to it, ask me questions as you lose it. You probably will lose it from time to time, and that's okay. Life is about learning, forgetting, and then hopefully you remembering it quicker than you learned it the first time. Okay. Why is this all so important? By the way, hormones all interact with target cells. What makes a cell a target cell from yesterday's lecture? What makes a target cell a target cell? Boom, receptors. So the receptors are going to be an important part of what you guys are learning. What makes a target cell a target cell? The presence of a receptor. No receptor for that hormone, well, that hormone cannot interact with it. End of story. Now, what is the one word that summarizes the entire endocrine system, that summarizes the function of the entire endocrine system? Communication. Awesome. Communicating. So that's what this page is all about. Communicating what? It's it's a list. It's a list. It helps to regulate the function of so many things. And non-essential, Tariq, not just essential, but non-essential as well. Hormones are pretty cool in their multitasking functionality. But you're not wrong in terms of what's on this list. Everything here is pretty darn essential. From growth and metabolism to blood, everything related to water, nutrients, ions, to how many red blood cells you have, how many white blood cells you have, digestion. It's going to be a big part of the digestive system. It's going to be one of the hardest things for you guys to learn in the digestive system is all the hormones involved in controlling digestion. Repro. Repro is regulated by hormones. You guys probably know this. To be an ionic portion of repro as well, and then last but not least, we'll talk about a little bit about circadian rhythms, not a whole heck of a lot. So just to go through this list, should you understand the basics of this list for the exam? Yeah. Am I ever going to say list the general functions? No, I won't do that. It's not my style. I'm not a lister. I don't really appreciate lists all that much. The other thing I want you guys to recognize is blood volume, plasma content, water balance, that sort of stuff. You'll be learning the basics of that in the endocrine system, this unit 1, but where you're really going to dive into plasma content, blood volume is in unit 2 when we're talking about cardiovascular. Does that make sense? Digestive processes will be heavily focused on in the digestive system. You won't need to know any of these functions for unit 1. Repro, again, we're going to focus on these for 4. You will need to know the basics of repro, but I will teach that to you. Hopefully I'll have time today to actually get to some of those basics. Circadian rhythms. Circadian rhythms are the easiest way to think about circadian rhythms is your body needs to know when it's night and your body needs to know when it's day. And then there are other rhythms throughout those cycles. Your entire body has waves of hormones. Waves of hormones that are regulated by the time of the day. I'll be teaching you guys about one in particular called growth hormone that are going to be regulated by another hormone called melanin. Melanin. Man, melanin regulates your overall circadian rhythms. No, melatonin, not melanin. I knew I was wrong. Caught it. Melatonin. Melatonin regulates all your circadian rhythms, but everything else is on a circadian rhythm. And for proper growth by the pineal gland, not the pituitary gland, but everything in your body that's regulating growth and development, well, that's controlled by growth hormone. Don't write any of this down. We'll get to it in just a second. But let me get you guys to think about this. It's only secreted during certain times of the night while you're asleep. So what's happening more and more nowadays, to me too, is that I'm staying up until 1 o'clock in the morning, 2 o'clock in the morning. So what do you guys think is happening to my normal circadian rhythms, growth, metabolism, and development? Is it improving or is it probably having some issues? It's just all of you that are parents, remember that when your kids are, sitting in front of their blue light at 1 in the morning, there's a reason why kids should be sleeping. They are in the critical stage of growth and development. And I'm not talking about muscles and bones, although I am, but the primary thing we need to worry about for growth and development are our brains. So just a thought, and we'll come back to that, but the hormones are really all about what time of the day it is. And if we're disrupting those patterns by not sleeping properly, then we will be disrupting our hormones, our normal hormone function. Love this that we already reviewed this. Y'all, through the semester, I will be asking you the following question. What classification of chemical is this? Can you please write that question down? What classification of chemical is this? What classification of chemical is this? So we'll be learning about all sorts of different chemicals, hormones, neurotransmitters, neurohormones, et cetera, et cetera, et cetera. And as we learn about them and as we review them, I'll say, what classification of hormone is this? Or what classification of neurotransmitter is this? There are three possible answers: steroid, biogenic amine, or protein. Steroid, biogenic amine, or protein. What must you know about it? Steroids are all made out of cholesterol. What did you just learn about cholesterol? Is it polar or non-polar? Remember, cholesterol is a type of fat. So is it polar or non-polar? Lovely, wonderful. So can it interact with water? Yes or no? Can it interact with water? Yes or no? You guys are already killing this semester. Great job. This is a huge part of your semester, is how the heck these things work. Okay, they cannot interact with water. We'll come back to why that's so darn important. But hormones like estrogen, progesterone, testosterone, cortisone, cortisol, aldosterone, that's your stress hormone. This one regulates salts. Estrogen, progesterone, testosterone, these are all growth, development, and repro, right? Your estrogen controls your bone growth, for instance, right? Which is one of the reasons women are prone to osteoporosis when they go through menopause. There's so many things that these different steroid hormones do. However, what do they all have in common? Every one of them has a cholesterol backbone. What does every single one of these hormones have in common? They're all made from cholesterol. And if they're made from cholesterol, it's called a steroid. So let's do this again. What do all of these hormones have in common? And if it's made from cholesterol, what classification of hormone would it be called? Lovely. So when somebody says they're taking steroids, does that mean anything? Kind of. It tells you that they're taking a classification of hormone, but it doesn't tell you what kind of steroid they're taking. Does that make sense? And we'll actually learn more about what that means if somebody's taking steroids. All of these, if you're taking an estrogen supplement, steroids, literally birth control pills, I'm taking steroids. You can say that because it's just estrogen and progesterone. So I always find that term funny. Yeah, and it's funny. Biogenic amines. So these are single amino acids that have been modified. That's a biogenic amine. They are amino acids. Remember, there are basically 20 essential amino acids that are the building blocks of all the proteins of your body. I hope that made sense. If not, I don't have time to talk about it right now, but I could talk about it with you later if you so choose. Amino acids are the building blocks for your proteins. So if you take a single, just like this green guy here, just the yellow guy, if you take a single amino acid and you modify it, we make a biogenic amine. Now, almost all biogenic amines are polar. Why? Because they're an amino acid. Amino means there's an amine group on one side. Acid tells you there's an COOH on the other side. So if you look at your entire protein string, one side of the entire protein string that's exposed to water is an amino. Amine means nitrogen. And the other side has this COOH, which means acid. We'll not deal with that too much. That's for a different class, a different place. What you need to know about that is that it creates charges, primarily a big, fat, negative charge. Biogenic amines tend to be negatively charged. due to their chemical composition. They tend to have a big, fat, negative charge hanging off of them. What does that tell you? Can it interact with water if it's charged? Yes or no? Loverly. Great job, y'all. Wonderful, wonderful, wonderful. You better believe it's a big yes. So most biogenic amines are going to be able to interact with water. There is one exception to the rule that will be very important for Unit 1 exam, and that exception is thyroid hormone. Due to the chemical composition of thyroid hormone, it will be nonpolar. Please write that down. All biogenic amines are polar except for thyroid hormone. So most of them, norepinephrine, epinephrine, melatonin, most of these biogenic amines are going to be able to dissolve in water, but thyroid hormone will not. We'll come back to the importance of that in just a minute. I promise. All right, well, if most amino acids are charged and proteins are made out of amino acids, what will all proteins be? Because we got this combination of thousands upon thousands of amino acids that are bound together. Will it be polar or nonpolar? Again, if most biogenic amines or just amino acids are charged, now you got just a big strand. Some of them are non-charged, sure, but as a general rule, big, huge, long strands of proteins have this, again, even bigger, even fatter negative charge hanging off of them. So proteins, can they interact with water? Yeah, definitely. Cool. ADH, insulin, glucagon, growth hormone, erythropoietin, all of these are just examples. Acetylcholine, which you guys learned about in AMP1, these are all going to be examples of protein molecules. So we'll come back to that in just a second. It will be important in the overall general function. Classification of chemical changes everything about the action of a chemical. So you better believe we're going to be learning about dozens upon dozens of different hormones, neurotransmitters, and whatnot this semester. In order to just know and understand how they interact with the cell, remember lipid versus water soluble. Remember nonpolar versus polar molecules. It will help you immensely in their overall function. Why? Because of where the hormone, where the chemical can interact. Something that is polar or water soluble, can it make it through the cell membrane? Yes or no? Don't stare at this slide, just work with me. Can a water soluble hormone make it through the cell membrane? No, because it's large and in charge. I love that terminology. If it's large and in charge, it cannot pass through a cell membrane. It can interact with water, great, but it can't pass through the cell membrane. So where will the receptors have to be found for that water soluble hormone? They have to be found on that cell membrane. Does that make sense? Lovely. And when they're found on the cell membrane, well, the cell membrane now is going to have to change what's called signal transduction, and there's going to be a process of cellular signal changes with secondary messengers and crazy systems that you'll have to learn about if you ever take a cellular physiology class. We won't focus on those things here. Versus lipid-soluble hormones, remember, they're trying to go from an area of high to low, just like water-soluble hormones are. They're all trying to go from high to low. But because of the chemical composition of the peanut butter here in the middle, can lipid-soluble hormones make it through the cell membrane? Yes or no? Sure. So where will the receptors be found for lipid-soluble hormones? Inside the cell or on the cell membrane? Perfect. And if I have an intracellular or an intracytosolic receptor, it changes the functionality of the response. You're going to see that the response now is internal. And we'll go through some slides. We'll help you understand that. Okay. I promise this will help again for the rest of the semester. That's the only reason why I spend so much time on these beginning slides. Reiteration. Redundancy is the key to learning. You'll hear me say this many times this semester, and I hope you agree. Sometimes it's going to feel pretty monotonous, but the idea is once it becomes monotonous, once you're like, Dr. H, we effing get this, then I've done my job, right? That's my opinion. Water-soluble hormones, are they polar or non-polar? And if I have a polar molecule, let's remind ourselves of some cell anatomy. If I'm making a protein, where does it all start? I know that's a really vague question, but I wanted to ask it this semester. Where does it all start? If I want to make a protein, where does it all start? Ah, good. Ribosomes is a good guess. Not all the way there. Amino acids, it's going to start before that. So let me just get to the answer. The answer is the nucleus. Good, Tariq. Great job. Good job, Becky. So if I have a nucleus, I feel like I got a little party going on here. Why? What's stored inside of the nucleus? What's stored inside of the nucleus, y'all? This should be one everybody can get. Yeah, the answer is DNA. 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 I'm pretty sure it's pronounced DNA. I got that from Zootopia. I can't take credit for it. So I got myself some DNA down here. Why do I care about DNA? Dr. H, just come on, man. This is like cellular biology. I don't care about it. This is one I find like 90% of students don't remember. What's the function of DNA? You guys can hopefully get a little bell rung by what Tariq wrote. What's the function of DNA? DNA stores information. It's going to start the protein synthesis process. So I like that you wrote that. It's not what DNA does, but it's going to help with that process. You're not wrong. Go ahead. You guys, Tariq's got it. I like to have you guys write even when the answer's already there because it's a lesson in learning. Maybe you're still not getting it from what Tariq wrote. Yeah, they're instructions. Proteins, well, they're big. And you can't store all of the proteins in your body for the whole entire life that you're going to have. So instead, we need to make sure that we have a big-ass cookbook, okay? This huge cookbook, huge. Imagine a cookbook that takes up the largest warehouse that you've ever seen. And imagine how small a cell is. And a single strand of DNA is about 3 meters long. Yeah. Now, imagine that you have about 10 trillion cells in your body. That's a lot of meters. I can't remember exactly what the total length is. It's something like, well, I'm not even going to try to guess the number, but you can go back and forth to the sun something like, I don't know, 30,000 times or something like that. There's that much DNA stored inside of your body. So how am I going to get that much or that length of material stored and packaged into such a small area? I'm going to coil it. And I'm going to coil it, 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 and I make it into a chromosome. Does that make sense? Does that ring a bell from some past cellular class? High school biology where you guys are like, well, why am I taking? I mean, it's pretty cool when you think about it with that in mind. It's so cool when you think about it with that in mind. And all of those chromosomes, right, when they're starting to go through cellular division, they end up looking like that cool X. And you got a bunch of those in your body. And a different portion of this chromosome, say for instance right here, has a very specific code of DNA. And that portion of that chromosome might encode for your hair color. Okay, it's going to encode for a very specific protein. And then each portion of that chromosome is a recipe for a different protein. And you got 23 paired chromosomes in your body. Each one of them contains thousands upon thousands upon thousands of different recipes for proteins. Big ass cookbook, right? Now, next lesson is that when we tell the DNA to go through a process called transcription, what happens next? Transcription, right? You guys don't need to remember it all. Transcription is going to occur, then translation, and then, that's a whole RNA process, and then going to a ribosome. Does that ring a bell? Ribosomes? And it's the ribosomes that build proteins. Yes, no, maybe so? This will not be on the exam. This is all review. And they do that by using amino acids. I do a lot of review here at the beginning of the semester to make sure everybody's on the same page. So they use amino acids to build big, long strands of proteins, ribosomes. There's both free ribosomes and fixed ribosomes. There are free ribosomes and there are fixed. What are they fixed onto if they're fixed? Anybody remember? Yeah, the endoplasmic reticulum, and they become, the rough endoplasmic reticulum. You guys all kind of on board with this? Maybe half of you, maybe two of you. Sweet. So now this information gets passed through this mess, these mazes, and they make proteins. Lovely. I love making proteins. This is fun. We got a problem though. We got a big problem. If I'm making proteins, again, are they charged or not charged? If I got a big old protein here, is it charged or not charged? Typically with a big fat negative charge hanging off of them. You got it. They're charged. Why is this important? Can a charged molecule make it through this cell membrane all on its own? Can it pass through a cell membrane? No, I got a problem, don't I? How am I going to get this protein out? No, we do not use protein channels. Proteins are too big. They can't go through the cell membrane, even if there's a channel for it. Well, there couldn't be, because if there was a channel big enough to let a protein through, everything else would go through it. It's too big. Ah, lovely. Not probably going to bind to a receptor. If it wants to get out of the cell, great answer though, Ashley. If it wants to get out of the cell, it's got to be packaged inside of a vesicle. Does that ring a bell? So if I have proteins, let's package it inside of a vesicle. Ah, yeah, Mel, we'll get there. One second. Where do proteins get packaged inside of vesicles? What is that structure called? Yeah, this is the Golgi apparatus. Again, if you don't remember all of these terms, you're going to be fine. And don't run away from the class. It's fine. The Golgi apparatus takes the proteins made from the RER, from the rough endoplasmic reticulum, and it packages them into vesicles. Now, here's the part that I find that students were probably never taught, is what is a vesicle made out of? What is a vesicle made out of? Vesicles are these small little bubbles, and you can think about them exactly like playing with bubbles outside. Melissa, so good. Great. You guys were taught. This is great. It's okay. Most people don't know this. Vesicles are made out of plasma membrane. It's the exact same material as your plasma membrane, phospholipids. So vesicles have a phospholipid. And they're not going to go through phagocytosis, actually. Phagocytosis, cyto means cell, phago or phago means to eat. So phagocytosis means this entire cell is going to go and eat something else. So we'll come back. So this is basically phospholipids. What did you guys just learn about phospholipids? Lipids. Peanut butter. Peanut butter. Does that make sense? It's a peanut butter sandwich. So if I take, using the Golgi apparatus, and I stick proteins and I package the proteins inside of this thing, can the proteins get out of this phospholipid bubble? Can they just freely move out? Hold on, no yeses, no nothing. Can it freely move out? Listen to the words I'm trying to say. These are proteins. And remember, proteins are polar. And if I have a phospholipid shell, I have a peanut butter core. So that is going to be nonpolar. Does that make sense? One step at a time, one step at a time. This is why vesicles can store proteins. Yes, no, maybe so? Help me out. Help me help you. Show me the money. I love vesicles, okay? They're a great understanding in how the body works. So we have a lot of proteins that are already created and they're stored inside of vesicles. Andrea, do you have a question? Again, I can't remember. It was Andy or Andrea or Andy?
I usually go by Andy, but Andrea's fine. I was just wondering for the phospholipids, is it a bilayer or is it a single layer? Because I was wondering if like which side the polar heads are on.
It's a bilayer. It's a bilayer.
Okay.
Because phospholipids seldom go into a monolayer. They really only do that in the digestive system that I'm aware of. Could be other places, but we'll learn more about the monolayers and how they can interact with fats inside of them. But if I'm going to have a protein on the inside, Andy, if I'm going to have a protein on the inside has to be polar. So it's got to be a bilayer. Make sense? Great question. Great understanding of how to ask the question. Okay, perfect. Now, here's the deal. This is a phospholipid. So what if I were to take this vesicle made out of phospholipids and I touch it to the bigger cell membrane? What did I teach you about phospholipids? Is they love each other, right? They want to interact with one another. True story? So if I take this vesicle and I touch it right here to the cell membrane, what it's going to do is it's going to fuse with that cell membrane. And I can't erase my cell membrane, but basically it would fuse, right? So if I were to draw this again, I would end up with a big ol' pocket. So it's fusing with that cell membrane, and as it does, it's able to release those proteins out into the IF. Does that make sense? And what's this process called? Again, based off of AMP1. This one here is exo, outside, cyto, cell, exocytosis, removing something from the cell. Exocytosis, one of the more important words for you guys to learn this semester. You'll be using this understanding of exocytosis many times. Exocytosis. This is what's important. If I'm going to take a polar molecule and I'm going to remove it from the cell, what process is required to remove that polar molecule from the cell? Again, I'm going to take a polar molecule and remove it from the cell, what process is required? Exocytosis. And one last bit, I don't know again who taught you guys AMP1 stuff and who went into how much detail, but what I'm going to ask you is what ion, what secondary messenger, what ion is critical in the exocytotic process? What ion is required for exocytosis to occur through the body? If you don't know, you don't know. I love that you guys are just guessing, and I love that so much. Never stop guessing. Use your brains and just have fun with it. So the answer is calcium. The answer is calcium. And when you write calcium, please always remember those 2+ charges to them. Pretty darn important that they have lost 2 electrons. Now, where I'm going to remind you guys this occurs, maybe this helps, is that if I had a neuron with a big long axon, and that axon ended up down here in the boutons, remember these little round terminal endings? Depending on who your professor was, you might have learned them as terminal boutons or terminal nerve endings, or I'm blanking on all the 1,000 other endings. I call them boutons because axon terminals, yeah. Boutons is like so much fun to say. Everybody say bouton, bouton. So, I mean, it's just, for me, it's always been easier to remember. And when you zoomed in on that bouton, it looked like this. And there were vesicles down here. And you guys learned that there, as an action potential descends, that action potential activates a voltage-gated calcium channel. Does this kind of ring a bell? And when you activate a voltage-gated calcium channel, now calcium enters, and that calcium causes exocytosis. True story? We will use this understanding of the body 1,500 times. Yeah, but it's not just acetylcholine. Remember that. Acetylcholine is just a protein chemical. So what if I'm talking about growth hormone? What if I'm talking about insulin? What if I'm talking about any protein? Yeah, but acetylcholine is not the only neurotransmitter. Lots of other neurotransmitters out there. So just keep that in mind. And the only reason why I'm mentioning this is because a lot of students learn so much about acetylcholine in AMP1 that they then come to AMP2 and they end up using acetylcholine multiple times on exams when they should not. Okay, acetylcholine is just one type of neurotransmitter. And remember, we're not just talking about neurotransmitters. We're also talking about hormones. We're talking about anything that's polar. All polar things are made because DNA is activated. And we get transcription, translation, ribosomes end up building these proteins, and then those proteins are packaged in vesicles within the Golgi apparatus. And then in order to get those proteins out, it needs to go through a process of exocytosis. And just one more time, what ion is critical for exocytosis? Good. So let's get back to the lecture. And what you'll notice is that we now can start to pull all of this information together. Water-soluble hormones. Look up above here. What is this water-soluble hormone stored inside of? Right here. A vesicle. So how the heck did this cell, this is a gland cell, how did it release that water-soluble hormone? What process was required for it? And what ion was critical in that process? Lovely. Now, what do we know about water-soluble hormones? They're all going to go from an area of high to low. Can they make it into the blood? Because they're interacting with water. Once they make it into the blood, can they dissolve in the plasma? Yes or no? Because they're water soluble. I mean, the name's right there. But because they're polar, can they dissolve in water? Yes or no? Don't be tricked. This is an easy answer. And boom, we've done our job. They're super easy to transport because now they're just going to be moving through the bloodstream. Now, here's the problem, is as they move through the bloodstream, they're going to be passed through the kidneys. They're going to be passed through the liver. They're going to be passed through the spleen. They're going to be passed through the body. And as they are passed through the body, there are chemicals that are digesting and breaking down these hormones. These hormones are not protected. They're just dissolved in water. And because they are so easily degraded, they have a very short half-life. A half-life means literally 50% of their life cycle. So for instance, insulin is a protein. When you inject a patient with insulin, it is only working really, really well within the first 4 to 6 minutes, because at that point, after about 4 to 6 minutes, only 50% left of it in the system. And then it's fully degraded within about an hour. Degraded, secreted, whatever the case may be, just depends on the hormone. Because it's not protected, you guys will see this a little bit better when we start talking about the lipid messengers. Because it's not protected, it's going to be degraded very, very quickly. That's going to be great knowledge when you guys make it to pharm and just in your future medical careers. Okay, Blackboard. Let me show you guys something else on Blackboard here. Lecture material. The next bit of lecture material I'm not going to cover in class because it's A&P1 material. And I have a narrated PowerPoint for it. So can everybody see this one here? It's called Signal Transduction Narrated PowerPoint. PowerPoint. .PowerPoint. Signal Transduction Narrated PowerPoint. I would like everybody to find some time to watch it. In order to watch these, actually, let me show you, because not everybody knows, oddly enough, which is fine. I shouldn't say oddly enough. I'll just open up a cardio because they're really big files. So when you go to download them, be patient. Have good internet access. Make sure you're on Wi-Fi. They're big because there's a huge audio file that's overlaid over the top of them. But when you go to use them, you can see now that this has a little star with a timer, star with a timer, star with a timer. In order to get these things to work as they're supposed to, you need to either click on slideshow up here and then click from the beginning, or you just come down here and it says slideshow. When you click on it, boom. You don't need to do anything from here. I'm hearing my own voice in my ear right now. But you just sit back and you listen to my voice. I try not to be too boring, I promise. I try to do the narrated PowerPoints the exact same way. Yeah, you'll be doing it for signal transduction, not for cardio. I just didn't have the signal transduction in my file, so I didn't open it. Does that make sense? And I didn't want to waste a bunch of time downloading it. Sorry about that. So yeah, please do that for signal transduction, and then you just sit back and you take notes. And the great thing about it is if you don't understand something, you just press back. You can go back one slide and you can listen to it again. So it's pretty helpful. Sweet. No problem. Is that clear? I want everybody to, this is going to be on the test, so you guys do need to know it. And that signal transduction is going to take you through all of these water-soluble messengers. Okay? In order for a water-soluble chemical to interact with the downstream cell, it's going to bind to a cell membrane bound receptor. There are 3 major types of cell membrane receptors. There are chemically gated channels. There are receptor enzymes. And the one that most students learn and then immediately unlearn because they just don't like it, we got GPCRs, the G protein coupled receptors. Do you guys have some small tiny memory that this existed from A&P1? And the answer is no. You don't need to write it in chat. Kinda. I love that answer, Becky. That's very honest. Tiny is all it takes, because if you have a tiny memory, you know that it existed, and you can kind of work off of it. These will be very important this semester, because what makes a target cell, y'all? Receptor. A receptor. You guys are going to be tested heavily on receptors this semester. Heavily. And depending on what type of receptor is activated, we can have a different cellular response. For instance, ions. How do we define an ion? What's the definition of an ion? Who remembers that? Maybe I should just say it because I'm kind of running out of time here. It's a charged particle. Good. So if it's an ion, it has a charge. Either a positive charge or a negative charge, right? Loss or gain of electrons is great. If anybody wants to go through how ions are created, again, office hours, just meet up with me. I'll make it happen. I can teach it really fast and really easy. But ions are charged particles. And if it's charged, can it just go through this nonpolar peanut butter core? Yes or no? Hell no, right? You guys learned that. I hope if you've learned nothing else from today, just have the basics of that idea. If I got something that's positive or negative, if it's charged, it can't go through a non-charged core. It can't. So ions are small. They're single or very small molecules like phosphate or bicarb. And these small ions can pass through a channel. So let's think about the functionality of this. If I open up a chemically gated channel and I'm moving ions, for instance, sodium. Sodium has 1+ charge on it. So if I open up a sodium channel and sodium starts to rush into the cell, what charge is going with it? What is the inside of the cell becoming? More positive or more negative? And didn't I just change the membrane potential? Didn't I just change electricity? Yes or no? So if I'm dealing with ion channels, I'm dealing with electricity. This is different than a receptor enzyme. Enzymes are activated. There's no electrical changes going on here. Here with GPCRs, they can do all sorts of shit. Okay, they can interact with ion channels or other secondary messengers and enzymes, and GPCRs are much more complex. They can do all sorts of stuff. So the cellular functions, depending on whether it's chemically gated, receptor, enzyme, or GPCR, each one of those responses can be different. So knowing the 3 major types of cell membrane-bound receptors is very important. Have I made myself clear on that? Take your time, study them. Do you need to know everything about them right now? No. Are you going to be learning about different types of chemically gated channels through the semester? Absolutely. Where have you already learned about a type of chemically gated channel? The neuromuscular junction, where acetylcholine was the ligand, and it opened up the nicotinic channel and allowed for more sodium to rush in than potassium left, and we changed the membrane potential at the motor end plate. Does that ring a bell? So you've already learned about a chemically gated channel, and you could use that knowledge to help you later. Insulin receptors are a great example of a receptor enzyme, and then GPCRs, those are muscarinic receptors and most cholinergic receptors. All right, so big portion of this I'm going to skip on over. This is also from AMP1, and it will be part of the cell signaling narrative PowerPoint. You guys will need to know these different secondary messengers. Just learn what they are for now. And then, again, when we make it to specific systems, we're going to be talking about specific secondary messengers. Like one of my favorites to test on is cyclic AMP and calcium. I tend not to test on cyclic GMP. This is for the vision and olfactory. Pretty much the only place that we see those used. And then inositol triphosphate and diacylglycerol. These, we will talk a little bit about these, but a lot less than cyclic AMP and calcium. All right. Again, most of that is all in your narrated PowerPoint. So don't panic. Take a deep breath. If you need help after watching the narrated PowerPoint, just let me know. Again, I have lots of hours for office hours. So there's no reason to ever panic. What about lipid-soluble hormones? Can they just pass through the endocrine cell that they are made from? Lipid-soluble. If they're non-polar, yes or no? Can they just pass through? They don't need to go through exocytosis, do they? There's no need for exocytosis. As they are produced, they will from an area of high to low. Process is called passive diffusion. They're just going to passively diffuse from high to low as they are made. Now, here's a problem, a big problem. They just got into the blood. Can they dissolve in water? Yes or no? Nope, 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 nope. So they need help. They need help. Help. Well, we got good news. We got helpers. Can proteins dissolve in water? Yep. So what if I had a protein that could interact with the lipid-soluble hormone, and that protein binds that lipid-soluble hormone and helps to transport it to where it needs to go? Sweet. I got an Uber system inside my body. Okay, this is an Uber for fats. Carrier proteins are the Ubers for your lipid-soluble chemicals. My mom, she cannot say Uber. She still calls it Erber. Cracks me up. I can't get her to change. It's always an Erber. Brian, give me an Erber. I don't know what that app is. Sorry. Erber? Moms, they're crazy. Yeah, get her a lift instead. She could probably say that. Although she'd probably end up saying lifed. So let me remind you guys that you already understand this. What carries oxygen around in the human body? Most people know this, even though they weren't ever, they haven't been taught it in A&P1 or A&P2 yet, right? Red blood cells, the blood, red blood cells, true story? Red blood cells. Most people know this already. Yes? No? Maybe so? Help me help you? We'll just go a couple more minutes and take a nice long break. Red blood cells carry oxygen. Why? Because oxygen, what did we learn about oxygen? Is it polar or non-polar? Oh my goodness. This is non-polar. This is not charged. Can oxygen dissolve in your bloodstream? Yes or no? Not very well. Not very well. So what does it need? What does this non-polar chemical need in order to be carried throughout the body? It needs a carrier transporter, a carrier protein. It needs an Uber or a Lyft. True story? And it's actually the hemoglobin found on red blood cells. That's the protein. The hemoglobin is a carrier protein. I said hemoglobin. I meant hemoglobin. All right. You guys good with this? It's technically even the heme group that's on there that does the job. But this is a proof in the pudding, real life. When I have a non-polar molecule, it needs a carrier transporter, a carrier protein for its job. So what did you just learn about testosterone? Can I just pump my body up with testosterone and it just works? Or is my body going to have to produce carrier proteins to carry that testosterone around? Yes, no? Sí or no? Carrier proteins. Gots to have them. So now let's just start to think about, yeah, we're okay. We're okay. We'll come back to this in just a minute. I want to give you guys a nice long break in just a second. Probably take 20, 25-minute break or so. So last little bit of probably learning for most of you guys. Greater than, I'm running out of room here, but greater than 90% of all blood proteins are produced by one organ. What is that organ? I'm going to put a blank. Again, most of you probably don't know this, but if you do, throw it out there. Ooh, I love that you answered bones, but that's not the right answer. Bones are making the blood cells. So that was your connection, Sabrina, and I love it. But the blood cells are actually not considered a protein. They have proteins on them, but they're not considered a protein. They're considered a cell. So good. Tariq, you got it. It's the liver. Okay, so I need you guys to learn this. Almost all blood proteins are produced by the liver. The way I'm going to teach, and by the comprehensive final, you will completely understand the importance of the liver, I hope. But what I end up saying about the liver is that the liver is always the bridesmaid, never the bride. Most people have heard this saying before. Always the bridesmaid, never the bride means that you're always doing all the damn work, but you never get the spotlight. Does that make sense? The liver does so much work. The liver is, I mean, if you got to talk about one of the most essential organs in the human body, it's the liver, because the liver helps your heart, the liver helps your blood, the liver helps your brain. The liver is going to end up, because of its detox function, because of its detox function and because of its ability to make blood proteins, the liver is just absolutely essential. So now let's talk about somebody with Hep C. So somebody has hepatitis, that means their liver is failing. And if their liver is failing, what's starting to potentially happen to that patient's blood proteins? Increase, decrease, no change. Oh, you guys got your first increase, decrease, no change question. You guys can just go IDN. So that's going to be a big part of your tests. Yeah, so if there's less blood proteins, one of those is a carrier protein potentially. So what's going to start to happen if there's less carrier proteins? What's going to happen to the transport of lipid-soluble hormones? Increase, decrease, no change. Houston, we have a problem. Your body's producing plenty of estrogen, plenty of aldosterone, plenty of whatever cholesterol-based hormone we're talking about, but that cholesterol-based hormone can't get to where it needs to go because that damn liver is failing. Now you got all sorts of problems. With that, let's come back at 55 after. Let's make it 57 after. Random, because I'm a random person. You're welcome. Get your bio, grab some water, grab a bite to eat. We still got a lot to do. We will take one more break before the end of class, but ciao. Hello, welcome back. I'm at 58 after, of course. Okay, I have one more slide that I really want to get to today, and then that's it. We won't have the longest day ever. A lot of these things like hormone interactions, control of the hormonal release, up and down regulation, these are individual concepts. Amplification, each one of these are found inside of that narrated PowerPoint. Just please, 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 I just want you to watch that narrated PowerPoint. They're really quick concepts, really relatively, not really, relatively easy concepts to understand. For instance, up and down regulation, it's all about receptors. If the cell membrane has more receptors, it's called upregulation. And the cell can determine how sensitive it is to hormones. If I have more receptors, am I more sensitive or less sensitive? And if I have less receptors, am I more sensitive or less sensitive? Now, let's say I have a patient that has hypercortisolism. Their body is producing too much cortisol, or hyperinsulinemia. Their body's producing too much insulin. What's the body gonna want? Does it like that? Does it want too much insulin? Does it like to? But now I got a problem. I got a disease, and it's producing too much cortisol. It's producing too much insulin. I can't do anything about that. But as a cell, I can decrease the number of receptors that are available for that hormone to interact with, trying to protect myself. So if I have a hyper disease, I want a down regulation in receptors to make it so that not quite as sensitive. Make sense? So my class will be a lot about learning a concept, memorizing a concept, but understanding how it can be used later. All right, the last big thing that I want you guys to know with me here today is the function of lipid-soluble hormones. First off, because lipid-soluble hormones use carrier proteins, they jumped on that Uber, the Uber protects them. So now that they are surrounded by a protein, they have a longer half-life. So if I get cortisol in my system, if I get aldosterone in my system, or estrogen or testosterone in my system, it's going to stick around for a longer period of time because it's not going to be as easily degraded. Does that make sense? Because it's being protected by a protein coat. Sweet. They're going to have to get through that protein first before it can get to the lipid. So, I mean, now we're looking at days for the degradation of a lipid-soluble hormone. Now, once those hormones are freed, they make it down to a target cell. Let me do this for the 1,000th time this semester. You guys will be doing this so many times. What makes a target cell a target cell? You guys can just get into REC or something like that. Yeah, it's receptors. But if I'm lipid soluble, where are the receptors found for my function? On the cell membrane or ICF? Cell membrane or ICF? Plasma membrane, PM, whatever you guys want to write. ICF is the answer. Sweet. Now here's the difference. What you noticed about these other ones up here is that these are all causing an immediate response at the cell membrane. Whereas if I get that lipid-soluble molecule, where am I at, down into the cell, now I can interact with organelles inside of the cell. Not at the cell membrane, but a direct interaction with the organelles of the inside of the cell. Oh gosh, I'm so sorry, guys. I do this after breaks sometimes, so please poke me. I just didn't share my screen. It's my fault. I just get going. This is like my number one issue probably is forgetting to reshare that screen with you. But you guys have been answering these questions wonderfully so far. So the rest of this lecture, this is what I was showing you: receptors, upregulation, downregulation, controls of hormonal release, the hormone interactions. These are all in that narrated PowerPoint. So that's what I was mentioning. You guys were still able to follow and answer my questions, which is great. But now I got a lipid-soluble chemical that once it makes it to a target cell, the target cell releases these little called paracrine factors, these little local hormones that tell the carrier protein to let go. And so now I have a lipid-soluble hormone that's going to go from an area of high to an area of low. So it's going to just passively diffuse through the cell membrane, because it can, and bind to a cytosolic receptor. Now, this cytosolic receptor is deep inside the cell, and it can bind to the organelles inside the cell. And the number one organelle that it interacts with, it's the nucleus. And what do we know the nucleus is storing? What do we have a lot of? Countless of hundreds of thousands of miles of? DNA. Good old DNA. And what do we know about DNA is that DNA recipes for protein. So if we activate or turn off DNA, we will be altering protein production. Does that make sense? You guys see the link? So what did you learn about all lipid-soluble chemicals, whether it is thyroid hormone, aldosterone, cortisol, estrogen, testosterone? Yep, they're nonpolar. But what did you just learn about their functionality? Every single one of those will be changing protein production, protein synthesis. Yes, no, maybe so? This is very different than membrane-bound receptors, which now change things at the cell membrane, immediate responses. They're interacting with proteins that are already made, not causing new proteins and new translation. Now, is transcription, translation, is it a really fast process, or is it a relatively slow process? Think of knitting a sweater, for Pete's sake. Somebody hand knits something, it takes a long time. Making new proteins in your body is slow. Days. It can take up to 3 days before you have a real impact. And I love this idea. So if you start giving a patient Cort, are they going to respond immediately, like before they left that doctor's office? No. So if they call you back the next day and say, I don't feel any pain reduction, you got to be like, just wait, you're going to be okay, kind of. Does that make sense? Testosterone, now we're looking at impacts over days. Aldosterone, this is going to be a really important one. Aldosterone is going to regulate salts by changing the number of receptors that are being produced. Remember, receptors are proteins. We'll come back to that later. But man, if you start altering anything in a patient's body, they are going to have a change in lipid-soluble chemicals. Well, now you're starting to look at potential side effects that are occurring 2, 3, up to a week later. And that's going to be a pretty cool little tidbit of information. Hopefully you guys can carry with you forever. All right, I need you guys to, I fail as a professor. I forgot to prep something. I was down playing with my 4-year-old and I forgot to prep something. So let me check to see whether ow, biscuit eater, just hurt myself. I'm a failure. Sorry, 1 second. I have something else open, so I'm not looking at the screen. Get rid of that. Let's read. All right, yeah, it's recording. It's continued to record from the last one. Let's see here. Endocrine lab models. Nope, I don't have it yet. Yep, I failed. Let me. This is going to take me about 1 to 2 minutes. Lo siento. I have to find a file that I stored away. Labs. Anatomy labs. Endocrine. You know what? Did I not make this one? Sorry, so for most of the semester, I have made what's called drag and drop. I thought I did it for this one, and I am not seeing it. Drag and drops are great. Why did I not do it for this one? I blame my administrative assistant. It's a joke because I don't have an administrative assistant. It's me. Okay. Well, I don't have it currently. It's not in any of my files that I'm looking for. That's interesting. But we will still go through and do something similar to a drag and drop. What a drag and drop is it's basically a PowerPoint presentation that you already have access to some of these. So for instance, right here. So if you go to the lab tab, lab material A&P2, lab 1, that's what we're working on today. There's endocrine lab models unlabeled. So you open that up and you're going to get, hopefully, a PowerPoint that's filled with a bunch of pictures of the models that we would have had in class. I think a lot of the A&P1 professors are going with something similar. But when we all started off, we didn't have pictures of a lot of these models. We just didn't have them. I thought I already mentioned that I was, but yes, I'm definitely recording. Anyways, when we all started off, we didn't have pictures of a lot of these models. So we were just sending students to go, search the internet. But the problem with searching the internet, and I still get students that want to do it this route. If you type in hypothalamus model pic, right, what you're going to end up getting is just a bunch of random pictures. Some of them are an actual brain model, and other ones of them have multiple things labeled. You don't want to look at everything that's labeled, right? You only want to look at what you want to look at. So what I provided for you guys are just blank models. And these are the blank model pictures amongst some other pictures, amongst some of the other pictures that I will use for your lab practical. Is that clear? There are other pictures of like the hemi head, the pancreas model, that sort of stuff. There are other pictures of these models out there that I can use, but I will use ones that are similar to these pictures. So what you guys are going to do is instead of having the actual models in front of you, you're going to have these pictures and you're going to label them. Is that clear? So for instance, here's a picture of a half brain. Now, what you can do is once you download these, just edit the crap out of them. So for instance, there are a bunch of different structures in the brain. I do not recommend that you label one picture with all 3 structures you're supposed to learn in the brain. Why? Because then you start getting really busy pictures and your brain has a harder time, a more difficult time learning specifically what you're after if you try to label one picture with too many words. You guys have all experienced this. True? You'll look at a model in the book and you're going to be like, oh no, are you kidding me? How do I get the information I need out of it? Well, it's easy, right? Just pick a picture, copy paste, and then you're going to make labels, So now you just go in there, you make a label. You want to make it as, clear as possible. I don't know what color is going to do that. Maybe this green. And then normally I'll actually make it a little bit bigger. So just change the weight of it so you can really see it well. So for instance, this guy right here, most of y'all should know it already. What is this? What am I pointing at right down here? So that's the pituitary gland. Good. Exactly. So that's going to be what you guys are going to be expected to do. Now, here's an even nicer way of dealing with this. Is now that I have this all set up, copy paste that, and now all I need to do is change where that arrow is pointing. So it's kind of a nice way to go about getting what you need. So this right here would be the pineal gland, for instance. So now we just type in pineal gland and we move this on over. So the pineal gland was also another one, hopefully, that you learned from A&P1. And then right above the pituitary gland, right here, well, that's the hypo, meaning below, thalamus. Below the thalamus was the core of the diencephalon, and right below that's the hypothalamus. So, boom. Now I've gotten 3 pictures labeled with 3 lab objectives. Does that make sense? Does everybody kind of know at least the beginning of what's expected of them? I got another brain model. What do I need to label? We're here to learn, right? So even though it might seem redundant, label them with the exact same 3 structures. Yes, no? Right? If you do it like this, now you've learned more than just the model, just one model. So if I were to, because you never know what type of model I'm going to throw at you. You just don't know. So when you look at multiple models and label multiple models, now you're learning. And that's what we really want to get at, is the learning process. All right, because does everybody see how they will be using these blanks that are on the blackboard? The blanks, unlabeled PowerPoints. Those are what I want you guys to end up taking, making your own, and then you're going to save it as a PDF. And that's what you're going to be submitting to me for extra credit. Everybody following along? This is your extra credit assignment. Create a study guide, which you should already be creating. I don't know why I have to reward students with extra credit for them to do what they should be doing anyways, but I've learned through the years, it's the only way to get some of you to make a study guide. So I am going to give you extra credit for doing, well, studying. There you go. And here's the even better part. As I mentioned yesterday when we were going through the syllabus, this is a group assignment. Lab groups. So what lab groups am I in, Dr. H? I don't know. Let's find out. Users and groups, lab groups, bulk actions. This is actually showing from previous. I didn't know that it would. Yeah. Add course, group assignments. Let's find out in terms of why. All sorts of things that I'm having troubles with today. Import, create, self-enroll, random enroll. Here we go. You guys ready? 1, 2, 3. It's a random shuffle. You don't know who you're going to end up with. Can I get a drum roll, please? All right. Damn it. Blackboard's always harder than I want it to be. Blogs, discussion boards, so you guys will be able to discuss with one another. Emails, so you guys will have emails. Journals, tasks, Blackboard, collaborate, wikis, academic. Pretty cool. You get all sorts of stuff. Number of students per group, we have 24, so we'll have 6 groups. That'll work. Distribute the remaining members among the groups. Yep. Won't be good there. I love it. You found the actual drum there. And then group information, we'll just go summer 2021. Can't believe it's 2021. All right, let's see what it comes up. Aha, and I'll have to erase these other ones here. But let's see what we got. So group 1 will be Ashley, Sydney, Melissa, and Jennifer. So go ahead and write your group information down. Remember, using these tools now, you guys all have access to your own Collaborate. So you guys can meet anytime on Blackboard. You just click on your Collaborate and you'll join your own Collaborate room. Pretty cool, huh? I think. So you guys can just do that. Now, I recommend one person in the group start a group email. So this group, for instance, here, you're going to select everybody, create an email, blah, blah, blah. And then in that email, you exchange your text message information or however you guys want to communicate, whether it's your own personal group me or so many different options nowadays. Yes, no? You guys see? Get it started. I'll give you guys 10 minutes after we get going here. 10 minutes, I'll give you guys 10 minutes to start to communicate with one another. But give me a second to get all the groups out. So group 1, Ashley, Sydney, Melissa, Jennifer. Oh, I should have just clicked the button. Anyways, group 2 is Veronica, Noah, Becky, and Andrea, but you wanted to go by Andy, I think. Oh, that was, yes, awesome. I try my best. So Veronica, Noah, Rebecca, Andrea, you're all group 2. Again, it's pretty easy because you just send out these group emails and you can at least get the process of communication going. All right, group 3. Sabrina, Sharon. Now, I think I asked this question yesterday, but is it Ruju? Yeah, that's right, because you said is Ruju. Ruju. Yes, perfect. And then Yvonne. Such a cool name. All right. Sabrina, Sharon, Ruju, and Yvonne. Group 4. Anna, Lidsi, Alyssa, and Trisha. Anna, Litzy, Alyssa, Trisha. Just go ahead and write down that you're group 4. Group 5. Alexis, Parker, Tariq, and dang it. I had it, Quinn. It's not Quinn though. Are you here today? I need some help. She's not even here today. So we'll call her Quinn for now, but I know it's not pronounced like that. Oh well, I get mad at myself. I love names, and I think names are so cool, unique, and wonderful. Let me go back because I didn't give that enough time. Alexis, Parker, Tariq. Oh, you already did it. Never mind. And then group 6 is everybody else. Alyssa, Ariana, Nicole, and Brianna, but you probably go by Bree. I think you even said that somewhere. Awesome. Nicknames are the hardest because all I have, unfortunately, I don't have a face. There's no like face-to-face interaction. Lab time's always fun together because we get to have that direct human interaction. Don't forget that Tariq also started a GroupMe. I saw many of you joined. Tariq, can you go ahead and relink that GroupMe when you get a chance? This is for the entire class, which is nice because if one person has a question, typically everybody has that question. And I joined that GroupMe. So if you guys want to ask a question to other students or to me, you can just text on GroupMe, okay? So what'll happen a lot of times, it'll be like, Dr. H, I forgot the deadline. And then other students can answer it too, which is nice because then we got this big group message going on. Okay, does everybody have their groups written down? Go into your group, and I want you guys to get that email out. I highly, highly, highly recommend sharing text messages. There are 21. Tariq, I'm guessing he's probably looking for the link. There it is. So you'll just click on that link and then join that GroupMe, Sharon. And then you guys can create your own like group 1, group 2 GroupMes if you want. Cool. Again, I'm going to give you guys 10 minutes. Somebody, one person per group or all people in the group, doesn't matter, send out an email. Start exchanging text messages, start exchanging phone numbers so you can get your group text going. And just be like, yo, this is me. I am trying to go into this program. I know it's not easy because it's like less personal by text, but go ahead and give it a try. So 10 minutes, so 35 after. Are you asking me, Alyssa? So if you go on Blackboard, so I'm not sending out the emails, not a problem. If you go on to Blackboard itself and you click on your group button, wherever the heck that's at for you, you'll just have access just to group 4 info. You'll end up with a page that looks like this, I think. And then you'll click on send email, Alyssa. So like your group is Anna, Litzy, Alyssa, Trisha, and then one person in your group needs to go and start an email chain. Does that make sense? Sweet. Litzy's on that that group email, sending out that group email, Alyssa. Is that, sorry if I wasn't clear the first time. No problem, no problem. Sweet. And then just check your email and then be able to respond probably with your phone number. And then, of course, texting is just so much more of today's everyday thing. Gmail would be my assumption. You could, I mean, I'm not quite, I guess you could, you got a group blog or a group discussion board. There are other routes, but I guess just determine that based on each group, how you're going to communicate. That would be my suggestion, because I don't want to regulate any of that. Does that make sense, Melissa? Just different people are so much more comfortable with different programs. Perfect. Yeah. Oh, yeah. It will be going to your email. Might just not be able to do a group message out. You guys will figure it out, right? You can just type it here if you don't mind the entire class having your phone number. You can be like, yo, group blankety blank. Here's my number. I am not tech savvy either, Bree. I am not. But that's why we got 10 minutes. And if you guys need more time for this, this is so important to be able to communicate with other people, to be able to communicate with groups of people. Even though we're strictly online nowadays and a lot of people work from online, you guys are going to be in the medical world. You will not be working primarily online, and you have to be able to interact with other people, be personable, be able to communicate. Smile even when you don't want to smile. Anybody ever been to a hospital and you hate the doctor, but not because you hate the doctor, it's because you hated the nurse? Like, we had the worst experience with our first child being born, not because of the doctor, not because of most of the nurses, but because of one specific overnight nurse. And that just ruined the entire experience for us. You guys are the face of the hospital. Don't forget that. Everybody needs to be on the same page. Yeah, our second kid was amazing. We had such a great overnight nurse. We were already stressed out for the first one, and then this nurse just didn't make it better. She was like, "You haven't done that yet?" Yeah, like, let her sleep. Exactly. My wife was woken, whatever. Anyways, we don't need to get into that. All I'm saying is you guys need to be somewhat amicable, able to communicate with one another, and so this will be an important part of that. And all I'm going to do, I know you guys are tired of hearing me peep into your ears, but I'm going to just put in a poll that says, basically, tell me when you're done. So it's going to be ready for the next step or whatever. I'll just say done. Don't click no. Okay, only click yes when you're done. Yes, correct, Veronica. Only when you're done setting up your groups. Once you guys get a group text message going, then I feel like the communication has started. If I don't force that issue, sometimes it just doesn't happen, and that just breaks my heart. Oh, let me, sorry. Try now. Aha. And if a particular person in the group, sometimes they can log in, but they Might be at work or they might be driving, something along those lines. So they can just be listening, but you know, not everybody can interact completely during in class. It is what it is with online learning. I get to teach AP2 back in person in the fall. I'm pretty stoked. Sydney, if you're there, if you can hear, Sydney, your group is looking for you. It will be due next Monday. Sounds right? Let me I printed it off. 1 second. Yep, the 7th. You got it. Try to make it as consistent as possible. Always the next lab day. No problem, Tariq. If it happens, it happens. Just keep reaching out to them. I mean, there's only 21 people here right now, so it's definitely possible that somebody wasn't able to make it today, and that might be a person in your group. Tis what it is. Go ahead and hit ready if everybody has responded that is here. And right now, again, just be patient. There are definitely groups that are still working on this. It is such an important part of your semester that I'm just patient with you. So give everybody some time to get it done. Looks like maybe just
2 more groups that haven't clicked that they're ready. All right, maybe. There's now 5 people that haven't clicked, but that could just be because those 5 people are some of the ones that aren't responding. Oh yeah, now we're getting somewhere. Now we're cooking with butter. Awesome. We're good to go. Now you guys as groups are all communicating. Such an important part of the semester. Please don't stop communicating with your groups. Okay? When you stop communicating with your groups end up telling me that happened. And when this happens every semester, and that's when you start losing out on study guide points, the extra credit, and lab report points, which are worth 10 points each. So keep communicating with your groups if you want, that good grade this semester. You're definitely going to need to do that. Okay, for those that are already really good communicators, great. But be patient with those that are learning how to do it better. Because online, working with groups, not everybody is the center of attention, the center of the spotlight. So please, please, please be nice to them. Sound fair? Okay. Thank you. 'Tis important. 'Tis important. I need to open one more thing here. Endocrine system study guide. All right. So there's 2 ways to go about getting ready for your labs. 1, open up your lab tab. Labs for Unit 1. Lab 1, you'll see that there is a doc and a PDF. Open up either one, doesn't matter, they're the same thing. These are your lab objectives. The lab objectives are what you need to learn. I don't know how to say this nicely sometimes. Stop studying crap in your books that aren't on these objectives. Stop giving me answers on the lab practicals that aren't on this objective list. If you give me an answer on the lab practical that isn't on this objective list, 1, I know you're cheating. I know you're using the internet while you're taking the exam. That's cheating. You guys are on an honor code not to do that. Just because it's open note doesn't mean it's open internet. Am I clear? You're going into one of the most honor-bound professions on the face of the planet. If you have to cheat to get there, I don't have any respect for you whatsoever. Sorry. Last semester, I had some big issues with cheating. We're having tons of online cheating problems because online cheating is a real deal. Stop it. Become the profession you want to become. Be honest with yourself and study more when you don't understand something. Reach out to me when you don't understand something. That's my job, is to teach you with passion. Your objectives list is not that long most of the time. This is a 1-page objective list. So if you're giving me answers that aren't on this objective list, I automatically have a huge red flag. Again, have I made myself clear? This is one of the few times through the semester you'll just hear the daddy voice turn on. Each one of you is fighting for a position in a future professional career. I get it, but earn it. Be the best you can be, not the best that somebody else is expecting you to be. Just be you. Don't cheat to get there. This list: hypothalamus, pituitary gland, anterior pituitary, posterior pituitary, thyroid gland, parathyroid, adrenals, adrenal cortex, adrenal medulla, pancreas, ovaries, testes. Very short list. And if you go to the PowerPoints, I always have this stuff there for you. Sometimes you have to search for it, true, but boom, right here. This picture right here has all of those organs that are on your objectives list: hypothalamus, pituitary, pineal, thyroid, adrenal glands with the adrenal cortex and adrenal medulla. In addition, that's not what you want. These are not the droids you are looking for. Where is it? Oh, that's right, I opened it as a PDF. In addition, pancreas, ovaries, and testes. So here's the other portion of it, and the hypothalamus up above. So, boom, they're here. Hypothalamus, pancreas, the gonads, testes in a male, ovaries in a female. Not too crazy, huh? For the beginning of the semester, you're going to get up to 3 extra credit points for putting together this study guide, and it shouldn't be too difficult to get you started for the semester. Let's look at some of these slides that I have for you. For instance, here, okay? So if I'm looking at this particular picture right here, this one on the right is better than the one on the left. I can start to identify, well, some of the major organs: lungs, heart, liver, the green thing is the gallbladder. This is the stomach, large and small intestines. Okay, cool. So what sort of endocrine organs can I start to see here? Well, the answer is none. Okay, some of these are kind of tricky. Are there tissues that contain endocrine cells? Absolutely, like the heart. Your lungs, guess what? Your lungs are one of those. They secrete quite a few different hormones that are important with COVID. You guys are going to be learning about them later. Stomach, small intestines, and large intestines, they're producing lots of different hormones. All right, up here in the neck, right here, everybody take and, while you're, go ahead and just like hum something, you'll feel the vibrations of where your larynx is found. That's your voice box. Now, just move your fingers inferior about 2 to 3 centimeters, and you'll start to feel some soft, fleshy tissue in there. That is your thyroid gland. Take your hand and put it in kind of a C shape and wrap it just gently around your neck. That is the approximate shape of that thyroid gland. It is like a gigantic C clamp. What that implies is the thyroid gland does not circle all the way around. Does that make sense? The thyroid gland is only going to circle, well, this is one side, the thyroid gland is cut off. It's going to circle around. This is the front of it. And on the back side of it, it's not going to be an enclosed circle. Instead, on the back side, there are little nodules. See this white bump? That white bump, that white bump, and that white bump. I only have these models here and these models here to test you on what's called the parathyroid glands. So the thyroid gland is right here. Again, there's the larynx, there's the cartilage, there's the hyoid bone. This is the thyroid gland. And if I take and I flip this portion of the model over, this is what you see on the backside. All of the red stuff here is the esophagus. So that's where food's going down. And this is the backside of this C-shaped parathyroid gland. So this right here, if I were to look at that structure, it's the I just said parathyroid, sorry. This is the thyroid gland right here. Okay, it's the side, this exact same side of the thyroid gland up above. But now you'll see there's 1, 2, 3, 4 nodules attached to the thyroid gland. And those are, who was listening earlier, what are these little 4 nodules right here? You don't have to spell it right yet. Just give it a try. Nope. Tariq, the answer is no. You do not have a baseline. This is purely as study guides go. You will submit them. I will look through them. If you want me to assess them, then you're going to need to make an office hour time with me, and I'll go through them with you. Otherwise, whether they're right or wrong, you guys will get your extra credit for it because you've created your study guide. But that's where the group dynamics comes into play. If 4 people are doing the work, four people probably aren't going to all get it wrong. Does that make sense? As they're going through and making these study guides up. And then you compare your answers, and that should fix any of these. Is my answer correct? And if you're ever curious, who can you talk to? Who do you call? Not literally call. Just throw me a text, yo. Just take a pic, be like, yo, Dr. H, I'm trying to find blah, blah, blah. I know how to use these text pics, no problem. You just I'll zoom in on something, draw an arrow, and I'll be like, there you go. Do not assess, we will get them wrong on the exam. Yep, exactly. Yeah, I think you were probably writing that when I was already answering that. You got it, man. If you want me to go through the study guide with you, we get on Collaborate, you share your screen with me, or you send it to me by PDF and I open it on my side. Boom, we can walk our way through it. Easy peasy lemon squeezy. It's a study dynamic that's unique to me, probably as a professor. I don't know how many others do it like this, but I want students to learn how to learn. I don't want to freaking have to, you're going to see a lot of review and teaching you guys how to study earlier in the semester, and then hopefully you can apply that. And then next semester, when you're in a different class, you're using the same study techniques and everything becomes better. Listen, I was a 2.6 student after my first 2 years of college. 2.6. That's a C, basically, a B minus. How did I get to where I am now is I had to make changes. And it was my physiology teacher, my physiology professor, Dr. Sanger, that changed my life. He taught me how to study. First time I'd ever been taught how to study. And I studied more with less time studying. And I was a straight A student from there, 4.0 from that point on. If you guys learn how to learn, all of a sudden school becomes easier and more fun, and then you end up like me, spending your entire freaking life in school because it's cool. It's fun. Anyways, sorry, that was a small tangent, but bear with me and you guys will learn how to learn, and it will change your entire life, I hope. Not for everybody, but for most. Sorry, that was a small tangent there, but does everybody kind of have an idea what they're supposed to be doing? This funny tongue-shaped thing over here is the pancreas. We got hypothalamus and pituitary glands here. I got a kidney, therefore I have an adrenal gland above it. So you guys can go through and look through these models and learn what they are. Most of the time I'll do like a drag and drop with you, like I said, but here's a model of the thyroid gland in front and the parathyroid glands behind. This really isn't the craziest of PowerPoints. Now, what you guys need to learn how to do is identify specific models. So for instance, this is a model. Some of you might look at it and go, WTF, Dr. H, what are you doing to me, right? This is the pancreas. This is a small portion of the small intestines called the duodenum, and this is the spleen. So we call this the pancreas model because it's kind of aligned around the pancreas in the middle. And then in this model, you can see the ovaries. And then you guys will be able to go through these as well. I promise it won't be too hard. What is hard? What I want to put more time and focus in is the histology. Who here hates histo? Go ahead, be honest. Be honest. Be honest. It's okay. I understand. Yeah, immediate PTSD in most students when I say histology. It doesn't have to be that bad. For you guys, I understand it's going to continue to be pretty bad because you have no context for why it's important. I didn't like it at first. Just I'm a normal student that way. I'm lucky because I've had lots of experience, so now I understand why it's important. Part of my job as a teacher is to try to teach you why it's important and how we can use it in the medical field. Now, as a nurse, are you going to ever be looking underneath a slide and identifying like chromaffin cells? No. But should you have a basic background understanding of what they are and how somebody, a pathologist, is going to use that information to diagnose a disease? Doesn't that make sense to know that, to have learned that somewhere? Yes, no? I'll teach you what chromaffin cells are in just a minute, but I hope you guys see that there is a rhyme to the reason of why nursing students have to go through this. And I mean, it's almost like a haze, right? You almost get hazed with histology. The one MD student in here, you got to know every single tiny little bit of this. And you're going to take other classes, of course, in your MD program. So let's get you started by giving you one of the best websites in the world for histology. It's one I found. Another professor has put it together. You don't have to pay anything. At least you didn't have to last semester, so I hope it still works. This is an Atlas of the Human Histology by a couple of professors. Looks like they're up at University of Minnesota. So let me link this to you. You guys open this link and then put it in your hotbar. You want this. You want to use it. It doesn't get much better than this website. Was anybody exposed to Slidebox, the histology guide in AMP1? My AMP1 students are, but I don't think any of you took me for AMP1. Yeah, Jen, that's great. That's awesome. It was sent out in an email. I sent it out in an email to all the rest of the professors. So I'm hoping that other professors are starting to use it. It is so darn cool. Like for instance, in AMP1, I think, epithelial tissue is always super hard for students. So when you click on epithelium, it teaches you what, things like the apical surface, the baso and the lateral surfaces are. So all these key words you learned or were taught, but a lot of students don't know how to use those. And then literally simple squamous epithelium, you can go in here, click common bile duct, and then you can go in and try to find simple squamous epithelium. But a lot of these ones, and we'll go back and out here, we're going to be doing endocrine glands. So for instance, when we want to look at the thyroid, boom, there's the thyroid. Don't worry, it'll make more sense in just a minute, but I'm just trying to teach you guys the program a little bit better. So this is a thyroid gland. And when we go to our list of objectives, ¿Dónde va? Hello? Hello? Are you there? Did I close it? I must have closed it. Well, that was very strange. All right, let me open it again. Easy. All right, so now when we go to our list of objectives, you'll see that the very first slide you're supposed to look at is thyroid gland. Okay? What do you guys need to see in the thyroid gland? You need to be able to find thyroid follicles. You need to be able to find follicular cells. You need to be able to find the colloid with thyroglobulin. And you need to be able to find parafollicular cells. Who here just heard those words? The words went in one ear, out the other ear. You're like, if you say so, Dr. H. Go ahead, be honest. It's okay. It's total gibberish. I know, I get it. Welcome to the beginning, but it will make sense, I promise. Well, I hope. I can't promise anything. Chief cells at the parathyroid gland, so forth, so on. So thyroid gland. So how can we use this amazing website. Look, thyroid follicle, colloid, follicular cells, parafollicular cells. Aren't those some of those words that I just said? What is a follicular cell? What is the thyroid follicles, I should say? Well, they define it right here for you. Spherical follicles of varying size in which thyroid hormones are stored. Sweet. Let's click on the hyperlink. Whoa. So you guys don't have to search all over the slide to find a thyroid follicle. You just click the button, follow the definition, and each one of these circles, see the pink goo on the inside? Pink goo, I call it bubble gum. Anybody see the bubble gum? Bubble gum, bubble gum, bubble, bubble, bubble gun. Bubble, 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 bubble. Yeah? Yes? In a dish. This is the beginning of the thyroid follicle. The pink stuff here is actually the colloid. So lumen of each follicle is filled with a gel-like mass called colloid. I'm not going to read this crap for you guys, right? It tells you right here. The pink stuff is actually thyroglobulin, which is a really important protein that helps to store thyroid hormone, which we'll learn about in lecture. So this is colloid, and it's found inside of a follicle. Wait a minute. Colloid with thyroglobulin. You guys starting to see that this program is going to help you be able to find these answers relatively quick. Way quicker than you would have otherwise. Yes, no? It's really helpful. Now, don't just rely on it. Try to learn from it. Yeah, exactly. So, Melissa, you caught it right on the nose. I do not give you pictures of histology because that would be insane. Right? There are thousands of different pictures that I can choose from. Millions. So now when you go to this website, where am I at? Here, right? Just take a screenshot and label it yourself. Yes, no? So this whole thing is a follicle. Do you guys see how there's a very distinct cluster of purple circles? Purple circles, yes, no? And they're very distinct. Sorry, I'm saying yes, no a lot right now. What is each of these purple circles? This is a lesson in a review that most students never learn. So let's do this. Know how to do what, Melissa? Yeah, if you're talking about a screenshot, Command-Shift 4. It's easiest way, and then you just drag your mouse. It kind of, creates a box around whatever you want to take a pic of. And then it'll just save right to your desktop if that's where you send that info. What are those purple circles? I'd love as many people as want to try to answer. Again, don't be afraid to be wrong. What are these purple circles? Purple circle, purple circle, purple circle. Nobody wants to answer. Any of the above, Tariq. Histology is a little bit more of a deep breath. Figure out the landmarks. I will teach you a lot of those landmarks, Tariq. I'll do my darndest. Ooh, chondrocytes. It's a good try, but there's no cartilage in the thyroid gland. It's just glandular tissue. And chondro means cartilage in Latin. Cyte means cell. So chondrocytes are making new cartilage. Does that make sense, Trisha? Great answer, though. Thank you for trying. I love that you tried. Parafollicular cells, great. Let's stop there. Para. What does para tell us? In Latin. I know, again, not everybody knows Latin. That's why we do this. Good, Alyssa, exactly. It means around or surrounding. Go ahead and write that down. Nope, it's not reticular. Good guess on that. Reticular looks like varicose veins. It looks like Lord Voldemort's head as he was like, coming out of being the weird little crazy alien fetus thing. Anyways, random. Para means surrounding. Follicular means follicles. So what's going to be surrounding the follicles is not part of the follicle. Do you guys see the reference to that word? Parafollicular are cells that surround the follicle. Are you with me on that? Peri is on the surface of, Nicole. Great question. Para means surround, like outside. Peri means on, surrounding but on. Great question. Keep working with me, y'all. Para means surrounding the follicle. Now, what I tried to show you earlier is that the follicle is these purple cells. We'll go around, these actually, the purple circles are not cells, but we'll get there in a second. And the pink bubble gum in the middle. All of this is one follicle. So parafollicular is saying it is surrounding the follicle. So they're the cells that are not part of the follicle. They are around the follicle. Does that make sense? They're not part of the follicle. So these here, this is the lesson in histology that most students just haven't learned because nobody had them stain their own slides to know what, these different purples and pinks and reds, and God, histology is so gross. The purples have a purpose. This is a particular stain. It's a type of a chemical that interacts with the nucleus, and due to the basic properties of the nucleus, causes the nucleus to turn this very distinct dark purple. So what are each one of these purple circles? They're nuclei. Are you guys with me? So if you can learn that about histology, it's super helpful. Because now I have one nucleus, one nucleus, one nucleus, one nucleus. Now, I'm going to have a single cell, and you got to just use your imagination for a lot of these, but the cell surrounds that nucleus. The cell surrounds that nucleus. You guys with me? As my mom would say, you hear my dogs barking? So there is one cell, and there's the nucleus associated with that cell. There's one cell. Now, these are cells that make up the follicle. So what are they going to be called? These are the cells that create the border of the follicle. They're part of the follicle. So look at your objectives list. I got thyroid follicle. What types of cells are going to make up the border of that follicle? And boom, right? Follicles are lined by a simple cuboidal to columnar epithelium, depending on functional activity. And it's these cells that secrete thyroid hormone. They're going to synthesize and produce and secrete thyroid hormone. They're pretty darn important. Is anybody here hypo or hyperthyroid? It's always a fun question. Typically have at least one student per class that is. Oh yeah, that has somebody. Hypo, Trish, yeah? Melissa, what is your husband? Hypo or hyper? Hypo. So how did, Trisha, if it's too personal, just don't answer, but how were you initially diagnosed? Were you gaining weight? Were you cold all the time? Melissa, you can answer the same for your husband. Were you just lethargic and tired and we're like, WTF, I don't know what's going on, I'm just tired? All the above. These are all not losing weight exactly and tired all the time. So you guys, based on those symptoms, doesn't it, anyways, we'll get back to this, but I love this idea. Now that you know some symptoms that come about because there's not enough thyroid hormone, you can start to understand what the function of thyroid hormone is a little bit better. And one of the major functions of thyroid hormone is metabolism. So if we don't metabolize properly, we're gaining weight, we're not burning energy quite as well. And if we're not burning energy, we're tired all the time. And boom, having the thyroid storm. Ugh. Yeah, I'll bet you had a lot of questions. Yeah, I love it.
Yep, yeah, Melissa, love it. Exactly. So listen, we got all sorts of different types of cancers that are out there, and cancer is the we'll talk about some tumors of the thyroid gland. We're going to talk about tumors of the adrenal gland. We're going to talk about a lot of these different types of tumors. But what cancer is the unmitigated growth of a cell. So now if I have a if I have a follicular cell that just starts to divide and my body can't kill it, and now I have massive amount of follicular cells that are growing into a tumor, into cancer, well, what's their function? They make thyroid hormone. So now if I have this unmitigated just growth of follicular cells, I'm going to have an excess of thyroid hormone. Make sense? Hyperthyroidism, lots of other nasty things that come about. Is everybody with me? I don't want to lose anybody. And this is not part of your lab practical, but it's helpful for your lecture stuff. We're going to be getting to it. I love to tie lecture with lab. I love to tie lecture with lab. Yep, you got it. Alyssa, we are talking about T3, T4. Absolutely. And we'll talk about why somebody that is hypothyroid might take just T3, they might take T4, they might take a combination of the two. We'll be dealing with it. Okay, colloid. What is the definition of colloid? Protein in a solution. This was A&P1 stuff. Most students don't remember that. Colloid, C-O-L-L-O-I-D. If you have protein in water, it's called a colloid. Protein shakes, colloid solutions. Your bloodstream has proteins in it. It's a colloid. These centers of a follicle are made up of a very concentrated protein solution called a colloid. What is that protein? It's called thyroglobulin. And thyroglobulin is going to play a very, very important role in storing thyroid hormone. We'll come back to that in lecture. Both T3, tri, and T4, tetra. All right, last but not least, parafollicular cells. Now again, you find a follicle. These parafollicular cells are the cells in between the follicles. They are around the follicles. Does the terminology work with everybody? Does it make a little bit more sense versus, I know a lot of students go, well, this is surrounding a follicle. No. That's part of the follicle. So those are follicular cells. Para are outside of that. Okay, cool. Was this helpful? I don't want to spend too much time because I don't want to bore you guys, but I want you guys to learn, and I know histo in particular students appreciate a little bit more help with. The purple circles, except for the ones surrounding the colloid, are parafollicular. And again, Becca, is it Rebecca? Becca? I think we asked this earlier. Becky. It's Becky. Duh. I know this. Becky. Remember the purple circles, those are nuclei. Those are nuclei. I know what you're saying. We just want to get a little bit more clear on our wording, especially in medicine, because a small problem with wording can make a big problem with understanding. And when you misunderstand what maybe a doctor is saying, now patients are going to be subjected to potential issues. So we really want to work on that, okay? And that's for everybody. So, boom. These are the nuclei of the parafollicular cells. Clear as mud? Mud's not clear. All right, cool. Does everybody feel a little bit better about the gibberish that you learned? Thyroid follicle versus colloid versus thyroglobulin versus follicular cells. Maybe it's a little bit less gibberish now and makes sense. All right, what's the next one we need to look at? Parathyroid gland. So we go over here,
We click back. We find the parathyroid gland. We click on it. What we need to be able to find are the chief cells. Well, here's the good news. Almost every single cell in the parathyroid gland are called chief cells. There are some other cells, oxyphil cells, clear cells, and adipose cells. You do not need to know those. You only need to know chief cells. And they are the loudest. They are the loudest too, Melissa. Some Seahawks fans might think they are, but nope. It's all about parathyroid. For those that don't follow football, just ignore me. Okay. So when I start looking at this, do you guys see how the purple, the staining is a different color? Let me go back. Let me go back because this is important. Thyroid, I'm going to just open this. Come on, open it in a new tab. So now when you start looking at the thyroid gland, do you guys notice these very distinct little bubbles? They look like pink bubbles, pink bubble gum. Yes, no, maybe so? The only gland in the human body that has these is the thyroid gland, no others. If you see pink bubble gum like this, it can only be the thyroid gland. Histology is easier if you learn landmarks. The landmark for the thyroid gland should be easy. It has follicles with colloid. Nothing else looks like this, not even close. Now, parathyroid, unfortunately, lots of things look like this. And as such, as a professor, I'm going to make it a little bit easier on you. Do you see the difference in colors? A light pink for the thyroid gland and a dark purple for the parathyroid gland. Yes, no? The other thing that's going to make it easier on you, and don't worry, I'm going to connect all the dots for you, is where do we find the parafollicular gland? Sorry, the parathyroid gland. Where do we find it? What's it attached to? Good, Alyssa. Does everybody kind of take note of this, please? I hope everybody sees that the parathyroid gland is attached to the thyroid gland. We'll come back to that. I got a really cool, just little side note on that I like to mention when I have chance, and we have a chance this semester. So what I will do to make your lives easier, I don't know why I'm going with, is that I will show you guys an image that always, always, always will also have, what do you guys notice about this picture over here? What's unique? What landmarks do you guys see out here? What are these? What are these whole units out here called? Good. The pink bubble gum is colloid. Follicles are the whole circle. That's kind of what I was actually looking for. Thank you, Ruju. I didn't do a good job communicating that though, so no worries. And if I see colloid, and if I see a follicle like this with colloid inside of it, what organ do I know it has to be? There's nothing else like it. No, not parathyroid. The colloid is part of the thyroid, but I think that was just a slip of the fingers. No worries. You maybe predicted a different question I was going to ask. So these are colloid. So this has got to be the thyroid gland. What do you guys see up here? Dark purple cells with no colloid. Yes? Dark purple cells with no colloid. Over here, it's not as dark purple. The whole staining is lighter, but you can see that there are follicles with colloid. So that's got to be thyroid. And here I don't see those follicles, but it's still physically bound to it. Here's even more zoomed in. Over here, colloid follicle. Over here, no colloid follicle. Over here, colloid follicles. Over here, no colloid follicles, dark purple staining. Here, tons of thyroid gland with a parathyroid gland attached to it. These are the sorts of pictures that I will use for the lab practical. Fair enough? That way you guys don't just get a bunch of purple dots that make no sense at all. You can actually make a contextual connection between the thyroid and the parathyroid. Fair enough? The large majority of the cells of the parathyroid gland are called chief cells. So if I were to throw a picture up like this for the lab practical and I pointed an arrow at this, don't just answer parathyroid gland. Read my question, because on the lab practical, the question will be either what gland is this or what cells are these? So if I write what cells are these, don't write parathyroid gland. That's not correct. The answer would be chief cells. Is everybody clear on that? Let me just slow this down. I love being a fair grader because learning hard enough, grading is hard enough, by the way. You can never be too specific for me. So what if I were to point an arrow at this and I said, what organ is this? Okay, maybe you're just like, I don't know exactly what there's other examples that are better than this, but maybe I don't know exactly what Dr. H is asking for. You can always write chief cells in the parathyroid gland, and you better believe I'm going to accept it. Is that clear? It's very helpful for students to be able to do that. Never be too broad unless the question is asking you to be broad. So if I said, what cells are these? And you said parathyroid gland, you would be wrong. If I said, what gland is this, or what organ is this, and you wrote chief cells inside the parathyroid gland, you would be correct. So a reward for having the very specific amount of information in there. Don't write everything under the sun. I don't need to know everything. In case we put an answer you would accept, but yeah, Alyssa, I grade them by hand. Alyssa, I didn't actually mention it yesterday. I grade them all by hand, so there's no like potential for computer error. You don't have to have that exact context. Anyways, you guys kind of get a pretty good idea of what picture I'll use for parathyroid gland and chief cells. They'll always have thyroid. Sweet. Okay. Next, adrenal gland. This gets a little bit more complex. It's not too bad. Not too bad. In fact, let's go to pancreas first. So we're going to jump to the pancreas slide. The pancreas is a funky little tissue. We're probably going to have to go. I don't know if there's going to be in the endocrine. Maybe it will. Adrenal. Oh, there it is. Lovely. We'll just pick, I don't know, any one of these. Doesn't really matter. Aldehyde. And probably the H&E is going to be the best. What I want you guys to look at here, you know the models that I've been showing you for the pancreas? They kind of just look like a weird, funky tongue. Yeah? Check it out. It looks like a weird, funky tongue underneath the, yeah, it looks like a flank steak, yeah. Okay. Pancreas has a lot of blood supply. So a lot of these big tubes you guys see right here, these big open spaces, that's blood. Those are blood vessels and arteries and stuff like that. But do you guys notice anything else landmark-wise? Let me zoom in just a little bit so you can kind of see this better. Is there something else just a little bit weird and as an outlier that just doesn't look the same as everything else? Yes, Tariq, by the way, to answer your question up above. The white striations, that's cool. Yep, the white striations are just in between the different lobes of the pancreas. There are actually quite a few organs that have these white connective tissue striations that divide lobes. Anybody look at this thing and say, man, this tissue has the measles, has chickenpox, has the mumps. What are those little white spots or pink spots? Yeah. Anybody see those? Anybody not see those? These are very unique to the pancreas. Only the pancreas has these little islands. Islands. They look like little islands in a sea of cells. These are pancreatic islet cells. Islet, right? Islet, island. In Latin, it just means island. I use island or, islet, islet. I don't use islet, by the way, of Langerhans. So instead of pancreatic islet cells, I use islet of Langerhans. But you guys can answer with either one on a lab practical, whatever you're comfortable with. So the pancreatic islet cells, that's what's shown on your lab objectives. This is one of the times where I'll tell you, go ahead and use islet of Langerhans. Most doctors still use islet of Langerhans. Langerhans was the name of the scientist that discovered them. What do these guys do? Why are they important? Why do I have them on my objectives here? One, they secrete hormones. But two, they secrete two of the most important hormones for metabolism in the human body. The islets of Langerhans secrete one hormone called, there it is, one hormone called glucagon. No, in my mind they don't, Tariq. And when you start looking at them a little bit closer, you'll see that colloid is just protein. So there's no nuclei inside of it at all. Whereas when we start zooming in on the islets of Langerhans, they're going to be little purple dots. They're going to be nuclei of cells that live inside of it. For Tariq, use whatever you want, honestly. For me, the islets of Langerhans have always been a real good landmark. So these islets of Langerhans, I'm just going to use some extra space that I have here. I don't want to use an entire page. These guys secrete two major hormones. There are a bunch of different cells inside the islets of Langerhans. There's alpha cells, beta cells, delta cells. But the two we'll be focusing on for lecture are alpha cells and beta cells. Alpha cells secrete glucagon, which helps regulate glucose. And beta cells secrete, no, anybody want to guess? Anybody want to guess a gander? Yeah. One of the most important hormones in the human body for medical purposes, one of the biggest taxpayer economic burdens in the United States, actually not one of this is by far the biggest economic burden for you guys as taxpayers, is treating people with diabetes. And it's this hormone right here that gets dysregulated in diabetes. So when you think of insulin, you're going to be thinking about energy, but when we want to look at how, there's going to be a lot of cool stuff about it, but in terms of the disease that's most often associated with insulin, we're looking at diabetes. Okay? There's your link. Just trying to give you guys, we'll talk more about insulin and diabetes in class. Everybody, what two hormones are secreted by the insulin and by the insulin? Son of a biscuit eater, I did it. First time this semester when I give the answer when I ask my question. What two hormones are secreted by the islets of Langerhans? And it's not glycogen, It's not glycogen. I got a few people that have written glycogen. Glycogen is something totally different. Glycogen is stored glucose found in livers and skeletal muscles. The answer is glucagon, not glycogen. Good job with those, though. Those words are very similar, and you're going to be dealing with those. Yep, both are meds, and very important meds, too. Okay, so the last one, and then actually we'll be done for the day, is the adrenal gland. Now, the adrenal gland, you guys are going to be definitely back to the point of, most of you guys will be back to the point of, I hate histology. There's a rhyme to this, though, I promise. But right now, it just looks like, oh, yep, there's a purple line, and there's some more like lighter purple, pink. There's darker purple. There's darker purple. What? There's a rhyme and reason to this. Do you guys notice relatively distinctly that there are basically striations of colors? And I already kind of mentioned those striations of colors. But you guys notice that, here's a real good example of it. I have a striation of color, another striation of darker purple, an area of lighter, an area of darker, and then an area of really dark. 1, 2, 3, 4, 5 very distinct layers. Yes? This is unique to the adrenal gland. So this whole thing is the adrenal gland. Bear with me. We'll do a couple of things to help you guys learn this and retain it. The outside layer of most of the organs of your body contain connective tissue that help hold it all together. This out here is called the adrenal capsule. Yeah, it's not keratin. It's all going to be made out of collagen. But this out here is called the adrenal capsule. And there's other, renal capsules. You got lots of different capsules for a lot of your organs of your body. But it's just connective tissue that helps hold it all together. So this very first band of pink is the capsule. So let's write this down. Let's write down the different layers. By the way, let's go ahead and do this. How many layers, how many different striations did I mention earlier? Good. So adrenal gland. We'll go ahead and say 5 layers. It's a 5-layer dessert. 1, 2, 3, 4, 5. From the outermost layer, we start with the adrenal capsule. Please do not just write capsule. Again, so many organs have capsules around them. You gotta know which capsule we're dealing with. 2, 3, and 4, and 5. We'll get back to that in just a second. But the first layer is the adrenal capsule. Okay, so this will be a nice list that you guys can study in just a minute. Now, the next layer, again, I know it's not super distinct, but do you notice that the cells here get a slightly darker purple tone to them versus down here where we start to see these relatively large, almost bubble-like structures? Yes? Where these little distinct. I know some of it's just like, if you say so, Dr. H, but if you really look at it, down here is very distinct relative to right here. And then when you look down here, just a little bit further, the structures change again. It becomes a darker purple. Up here is a little bit lighter. Down here is a little bit darker. And these circles get smaller and smaller as we go. Our striations. I was going to say something and then I forgot. Okay. I'll come back to it some other time. I'm sure I'll remember. Now, what I love to use for learning is I don't try to memorize everything at once because, man, deckages make life pretty tough. in my opinion. So what I'm going to do is I'm going to find an abbreviation or a mnemonic or a list or something that will help me remember it, flowcharts, and then I'll be able to go from there. So the next layers, 2, 3, and 4, are all going to be called zones or zona. So we just start by writing zona. So all 3 of those are zonas or zones. And now what I want you guys to remember is an abbreviation. GFR. This will be an important abbreviation when we get to the renal system, although it has nothing to do with the adrenal cortex, but it's still an abbreviation you're going to use again this semester. So it's a nice abbreviation for you guys to learn. Can everybody go ahead and write in chat? GFR. GFR, 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 GFR. If you guys can learn and memorize this GFR, everything else becomes a little bit easier, because now on a lab practical, if I show you a picture like this, and then I have an arrow pointing, say, to this layer, what you do is you go capsule, G, F, R. Does that make sense? And I will show you guys something that looks a little bit like this. So it'll have all layers of the adrenal gland in the histo picture, so you can count them. Capsule, G, F, R. This is the zona fasciculata. So F stands for fasciculata. What a weird word, right? But if we remember G, F, R, remember, this is an open note. So you can have your objectives list open. And we can go G, F, R, therefore we could write zona fasciculata. Is that helpful? The G stands for glomerulosa. The S stands for fasciculata. And the R stands for reticularis. Crazy words. If you memorize GFR, you'll be fine. Again, because it's an open note format. If it was in class, you'd have a little bit more learning memorization to do. Last but not least is the adrenal medulla. The adrenal medulla is the core, which is bizarre because all of this out here is called the cortex. So the 3 zones together, this is the adrenal cortex. So the way I'll have my lab practicals is I'll say, what region? Let me go back to the picture. If I pointed an arrow here, I would say, what layer is this and what region is this found in? The answer would be zona fasciculata in the, or just zona fasciculata adrenal cortex. Make sense? And you can see that's how this is organized over here. Capsule versus cortex versus medulla. Now the medulla has a cell in it that's important. The large majority of the cells found inside the adrenal medulla, these dark purple stains here, these are called, yeah, sure, Alyssa, sure thing. I'm going to go back to it in just a minute. So let me just go ahead and do this. These dark purple cells of the adrenal medulla are called chromaffin cells. So let's go ahead and write that in. The adrenal medulla is comprised primarily of chromaffin. I do not know why they're called chromaffin. Chroma means color. I don't know where the rest of the Latin context for chromaffin comes from. I wish I did. However, what I'm going to go ahead and tell you about these chromaffin cells is part of their function. Their function is one you guys should remember from AMP1 in terms of not, because you never learned about chromaffin before, but you did learn about the sympathetic nervous system. And you learned that the adrenal medulla secretes two major neurohormones, norepinephrine and epinephrine. This, what did I do? EIP? No. Epi. This is adrenaline, okay? It's the combination of these two things. 60% epinephrine, 40% norepinephrine. This is adrenaline. Anybody ever had an adrenaline rush? Whether it was from a car accident or jumping out of a plane or what sort of feelings do you have when you have an adrenaline rush? Are you calm? Are you chilled? Are you like, yo, man? Are you like wired? Let's do this again, especially if you're jumping out of a plane, right? Not chill. Adrenaline rushes are all about the sympathetic nervous system triggered heavily. It's an emergency response. I was chased by a moose around a tree for about 6 hours once. Anybody ever watched that TV show called Northern Exposure? So this moose was one that I helped care for. It was actually down at Washington State University, not up in Alaska. So this damn moose right here, this damn moose chased me around a tree for about 6 hours. And nobody knew that it was happening because he'd busted out of his gate. He'd literally lifted like this almost 1,000-pound gate. He'd lifted it off with his horns, with his antlers. They don't have horns. And I basically got him back into his pen area, and he just chased me around a tree. Talk about an adrenaline rush. So how was I able to stay on my toes for that long and not just give up? Adrenaline. Yeah, you got it. So this is adrenaline. Now, let me get you guys to think, what if I had a tumor of my chromaffin cells that caused an overproduction of adrenaline? Cancer's bad, yo. Is that patient going to be chill? Or are they going to have anxiety all the time, hypertension, vasodilated eyes, decreased digestive system because they're constantly in a fight or flight, heart rate, tachycardia? You guys see what I'm saying? Kind of, ish. We'll be dealing a lot more with this, so don't worry. This is not the only time you're going to see these sorts of questions from me, but I hope you guys are going to start to feel here just in the first week how my teaching style will be leading you out for the rest of the semester. It's all about understanding context of how things go together. Do I have this information somewhere else, Dr. H? You better believe I do. I would always do that for you. So in endocrine 3, here's the thyroid gland. Everybody see the colloid-filled follicles? Colloid, follicular cells, parafollicular cells around it. They're all about T3, T4. Here's how it's regulated. We'll deal with this later. Here are parathyroid cells. Again, I won't show you just a picture of a bunch of purple blobs. That wouldn't make any sense. Boom, here's the adrenal gland. In this picture, the very first layer is the capsule, is the dark purple here. But I love this picture over on the right. Everybody, can you see the dark purple capsule is blue? And now I got 1 layer, 2 layer, 3 layer, and then another 4. So really it'd be, let me put in some text boxes. So based on what you guys learned, 1 is going to be capsule. Yes? 2 is going to be zona glomerulosa, GF. 3, zona fasciculata. R, zona reticularis. Come back. And then 5,
adrenal medulla, and to test your short-term memory, what cells are found inside, what's the primary cell found inside of the adrenal medulla? Not chief cells. Good try, Veronica. That's a memory from a little bit earlier. Chief cells are from the parathyroid gland. Great try. Here we got chromaffin. And for anybody that's ever heard of a pheochromocytoma, that is a cancer cell of those chromaffin cells, cause them to hypersecrete. Here's another picture of pretty much the same thing. There's the zona glomerulosa, zona fasciculata, zona reticularis, a little bit less distinct. But what I love about this picture is it starts getting you guys to think about the different hormones that are produced. Colored muffins. Yep. Colored muffins? I do love muffins. So the zona glomerulosa, you do not need to know this for lab, but you will need to know this for lecture. The zona glomerulosa secretes aldosterone. Any chemical that ends in ONE means that it is cholesterol-based. It's a steroid. Aldosterone is a steroid. So is it polar or non-polar? Good. Good. Great short-term memory from today's lecture. Cortisol is also known as cortisone. Oh, so what do we know about cortisol? Cortisone. It is also steroid non-polar. Androgens are sex hormones. Testosterone, progesterone, and estrogen. Those are all steroids. Steroids, those are all polar. Okay, testosterone, estrogen, progesterone. Those are all steroids. So what did you just learn about the adrenal cortex? The adrenal cortex always secretes steroids. And all of these steroids are non-polar. See? Oh no. And that should make sense to you a little bit better after today's lecture. Now we know that these steroids, these hormones, are going to be causing DNA to produce more proteins. See the connection? You learned a bunch of puzzle pieces, and then you put the puzzle together. That's the hope. That's always the plan. Doesn't always work, and then, you know, I'm here to help. I'm here to help. Got some tables to put that all together for you. Zona glomerulosa, aldosterone. Zona fasciculata, corticosterone, which is the same thing as cortisone. Zona reticularis, adrenal androgens. Androgens are the base for all sex hormones. So an androgen is actually a testosterone, and then testosterone is changed into estrogen and progesterone. So it all starts with an androgen, which is testosterone. That's for girls and boys. And we're done. How do you guys feel? Is there anything else that you really need me to go back over? Are you guys ready to go and start to work on these labs as a group? I'm going to show you guys how to submit your answers real too here, but is there anything else you guys want me to go over with you? The objectives list we've covered. Can I get a nah? I don't know if I'm there. Am I lagged out? Sweet. Okay, cool. So the last bit of this is, how do we submit our study guides? Please pay attention, because this is not, there's no easy way to do it in the Blackboard we have. ACC has a cheaper version of Blackboard. There's a more expensive version of Blackboard that, you know, I would be able to bypass this crazy thing that we have to do, but there's not.
So there's an entire tab down here called Lab Group Study Guides. That's where you're going to be submitting all of your study guides for the entire semester. The way these study guides work is that you will be submitting them as a group. I'm not changing the due date because we don't have big free storms and anything else that's going crazy. You're going to be uploading your study guide, or you can insert your link, but I'm going to change that and I'm going to just say as a PDF. I found that this is easiest for students and for me. Make sure, as I mentioned yesterday, that you tell me who participated and who did not participate. You guys clear on that? And then this is all going to be June 7th at midnight, so it'll be due after. Now, it says it's worth 0 points because it's extra credit. So I will assign points to you. And when I do, everybody will get an additional 3 points added to their grade. Does that make sense? Just extra credit. You have unlimited attempts to be able to just go in and submit, resubmit. You know, again, I'm not trying to punish you guys with this. I want everybody to get 3 extra points. Yeah, exactly, Brianna. 1 second, 1 second. Great question. Availability, make it available. Okay, submit. All right, so now it's available for you guys to do. When you go to submit, you're going to click on this. As I was mentioning yesterday, you hit write submission and you say, so-and-so submitted, these people contributed. You add, by browsing your local files, you'll add your PDF, then you hit submit. 1 person per group needs to do this. That's it. So just communicate who will be submitting. One of the biggest problems groups have had is not communicating well. So 1 person or 3 people think that 1 person is submitting, but then that person doesn't. Communicate that the submission is done. Fair? Thumbs up? Veronica, spot on. The objectives list is your list to study. These are the words you must know, must be able to identify on models, histo, and then through the semester you'll have other things you need to be able to identify. But that's what's going on here. The study guide is just the study guide. It's a group study guide, as many brains that come together to put a comprehensive list of these pictures that are labeled. You can put in extra text, like I've had people that build these really cool PowerPoints. So say, for instance, they'll add a new slide and they'll actually do something more like this. So then they'll say, blah, 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 blah. So they'll actually give an, you know, what is this that I'm even looking for? So hypothalamus, and then they give a definition of what the hypothalamus is. Thalamus. And then they'll actually go through and say, blah, it does this, this, and it's really helpful. Totally up to you guys. Depends on how much work you want to put into it. Yes, no? The only thing you will be graded on is that the objectives list is covered. I go through, if everything on the objectives list has a slide, you get a check mark from me. You get your 3 points. Whether it's right or wrong, as long as you guys tried, you'll get your 3 points. And then you just hit submit. Lab reports will be a little bit different, but boom, we got our first thing up and running. Let's see, one last thing, labs for Unit 1. Do you guys see hematology lab safety presentation here? It's the second thing on the list. And if you look on your schedule, blood safety training is for today's lab. We're not going to do it together as a group. This is super duper simple. You open up the PowerPoint presentation, and then you just follow it. Boom. What do? Yeah, print out that handout. You don't have to print out the handout anymore. Read each one of these slides, okay? The slides are going to give you a brief understanding of lab safety. Are we doing any labs? Do you really need to worry about biohazards this semester? I wish. But we're not actually doing any physical labs, are we? Should you still be trained in how to handle biohazardous material? Got to learn it somewhere. You don't want to learn it like past the point of when you need to use it. And so we're still, as a biology department, we're still requiring students to have a basic understanding of biohazards, okay? Go through this study guide, and then your guys' very first quiz is called a hematology blood lab safety quiz. Let me edit this real quick. Once you get done, or even while you're doing it, as you're going through the PowerPoint, you have unlimited attempts to get 100%. This will be due on the 7th. Oh, display after. No, display after today. This will be due on the 7th at midnight. Once you get done with the PowerPoint, go in, take this quiz. It's super duper easy. One per person. This is not a group thing. You're including this grade in the scores and whatever. Boom. So now you guys have a hematology blood lab safety quiz that's available for you. And actually, I need to edit this real quick. It's worth 5 points. Is everybody clear on what's due by Monday? You have 1, a lab group study guide, and 2, you have in the exams and practicals tab, you have a hematology blood lab safety quiz. And that's it. I'm done. Are there any questions? Hope you guys are appreciating the format, having a little bit of fun. It will start speeding up here soon. Reach out to me if you have any questions. I will see you tomorrow morning.